So when your parents ask me, Adam, take those out of your ear. When they ask me what's going on, I'm going to say, refer to this day's notes and take a look and listen to what's going on in this class. Life. Obviously, you'd write the date, assuming that you don't want to, again, be asking, when did this happen? So life, yes, I've said, when I speak, I write it down. If you're listening and looking, it makes sense. I'm just letting you know. You have to connect the two. You can't be looking to the side or talking to someone aside or rolling yourself across the room and then look on the screen and then raise your hand and say, what's going on? Focus, stay together. Life. The characteristics of life, we talked about it. What are they? Homeostasis. Life keeps homeostasis. That's right. Metabolism. For those of you that don't remember, metabolism is processing energy. Processing energy. So homeostasis, metabolism, what else? What else does life have? In general, what else does life do? Say that again. Reproduction, I heard, is correct. Life reproduces. It makes more copies of this. So I think everybody agrees with that part. There shouldn't be an issue there. No, you're not. It's just doing its thing. That's right. Life evolves. Now, I'm going to tell you that this might be an issue for some people. I'm going to say it again. You just have to understand that that's one of the characteristics listed. Do you have to believe in evolution? Regardless. I mean, evolution is a fact. The part that is confusing to people, or parts that people argue, is did humans evolve? The way that evolution is defined, it's literally a fact. Like we can look at evolution in a test tube and with bacteria, and we can count the genes and the changes, and it's undeniable that evolution exists at the micro scale. The big question that people keep going back and forth is, did humans evolve, etc. We don't have to get into that conversation in this class. You just have to understand what evolution is, okay? So one of the characteristics of populations, not an individual. Let's be clear on that. Individuals do not evolve. Populations evolve. Populations. But life does evolve. All right, what else? Yes, but we're not going to list that. But yes, life dies. Life passes on inheritable information, right? Inheritable information in the form of DNA or RNA. Does everybody agree? Again, very provable. We can take your DNA from your body and we can compare it to your parents. You see that you literally are half your DNA came from mom and the other half came from dad. That's the only place you get DNA is from your ancestors. Information, I'm sorry. I'm writing quickly. I apologize. We have to get through this quickly. There's a lot to do. So inheritable information. DNA or RNA is how we... Then we talk, if you did the characteristics of life and looked at the answers that I posted, you know that life exchanges energy for order. All right. The universe is in chaos and is consistently moving towards disorder. Life does the opposite. Life takes this random set of mixtures and collects them and organizes them. Life is ordered. Life is ordered. It's organized. Which brings us to one other characteristic of life. All life, according to cell theory, all life is made of cells. Now, this is an arguable point. And the arguable point is some people consider viruses alive. They are not cells. Viruses are not cells. You'll understand more what a cell is as we go on, but life is made of cells according to cell theory. Now that brings us to the idea of a theory versus law. We have natural laws. Natural laws are observations of phenomena. Phenomena is another word of saying is happening. Like I throw an apple up, it comes down. That's a law, the law of gravity. Every time something goes up, it comes down. That's a law of gravity. I mean, we can see it. Now, how do we explain it? Through the theory of gravitation, the gravitational theories of Newton is how we explain it. But every time you throw something up, it comes down. That's a law, right? You can see it. You can watch it happen. If I drop something, it doesn't go floating up, right? So that's a law. A law is just are things that we see. Now, theories are our explanations of happenings. How do we explain what's happening around us? We use these things called theories. Theories are very powerful. They allow you to have synthetic shoes. Those shoes you wear are made of polymers that didn't exist in nature before humans made them. Electricity runs through those wires because of the law, because of our theories about how electricity works. Everything around you is made using theories, these explanations. So understand, some people think theories are weaker than laws. Well, I don't know about weaker, but theories are how we explain what happens, and laws are the happenings themselves. All right? So when we see life and we look at all life on Earth, we decided that all life is made of cells as a basic unit, and that's what we call cell theory. So that laws, it's difficult to say laws are wrong, but theories are constantly being proven wrong. In fact, we go ahead and assume that almost every theory we have is not complete. We assume that somebody's going to discover something next year or in 100 years that says, oops, look at what we were thinking. For instance, do you know the Middle Ages? Do you know how what they would do when you had a fever? In the Middle Ages, they thought there were 5 elements. They thought it was air, the Chinese thought 5, I think maybe, I don't know, you can do the research here, 4 or 5. Earth, air, fire, water, and chi or spirit, depending on where you are in the world at the time. So these 5 elements were it. That's all there was. So in Europe, instead of chi, they called it the humors. So those 4 plus the humors. So if you were sick, you had a fever. what did you have too much of? Which of the 5 elements did you have too much of if you have a fever? Fire. You had too much fire. So what they would do is they would cut you and bleed you. Well, they got cooler, but they were dying. But they didn't know. The theory was that you were getting too hot because your fire element was too hot, was too much, so they would bleed you in Europe. Sometimes they would put leeches on you and bleed you that way. Sometimes they would bleed you by cutting your veins and watching you bleed, then seal you up to hold it. Don't let you go too much. They would time it. They would take literally a pan full of your blood out and throw it out the window. And that was their idea of medicine. All right? Yeah.
Isn't that what they did to George Washington after he caught a fever?
I'm not sure if that happened with George Washington or not, because by the 1700s, our idea of medicine was better.
I think he had kind of failed.
Huh?
It was one of them.
I think it was George Washington.
It could have been. I don't know, to be honest.
They killed him.
Yeah, I don't know. But here's what I'm going to tell you. The theories change. As we get more information, our understanding improves. And we get better at what we do. I don't think anybody would argue that medicine's come a long way from bleeding people and putting leeches on. And by the way, leeches are still a good solution to things like bruises. They can take out the coagulated blood. They have anticoagulants in their venom, and they also are painless because they have. In fact, if you get a leech on you, you won't even know it's on you because they numb your skin. So they're very good at what they do. Medical leeches, though, they're grown in a lab. They don't go out in the swamp and collect them. They don't want anybody to get sick from them, right? So leeches are they're natural, and they don't harm you for the most part. All right, they're not like mosquitoes. To my knowledge, they don't really spread disease. All right, so I'm not saying it's good for you to get a leech on you, but I'm saying we do use them, like when a bride gets a black eye because she ran into their door or something, and she doesn't want to take pictures of a black eye, a swollen joint or whatever. They'll put leeches on the eye and drain the sac so that the black eye just goes away. The damage is still there, but the black eye is coagulated blood. You know that? Well, the leech has anticoagulants, and it'll actually suck that useless blood out of you. All right. So, anyways, old theories were wrong, but they were looking at phenomenon, and they were trying to explain them, and they did the best they could. All right. So those are the characteristics of life. So if you're going to take, I'm going to focus on this disorder, the word called entropy. Entropy. Remember, entropy is disorder, a measurement of disorder. The higher the entropy, the more disorder. Life does the opposite. Life takes energy and changes disorder to order. In order to do that, as far as we can tell, you need to create something called a cell. Because if the universe is mostly disorder, if the universe out here is mostly disorders, random material floating in every possible direction, then we have to be able to organize things in here. Inside this space, we call it order. Now, where have you heard the word cell? You've never heard the word cell before? 3 minutes ago. Yeah. I meant, what was the first time? Have you heard it anywhere else? Excuse me. Let me rephrase that. Okay. Have you heard the word cell anywhere in our language, used anywhere? Have you watched a show where they mention the word cell? Yeah.
Dragon Ball Z.
Okay. Have you ever heard of a prison cell? So what is? Okay, a cell phone. All right, that's good. I'm not sure how cell phones work into this, but prison cells. What is a cell? a room, right? A room. So a cell is a compartment room, space that's separated from other things. You need to separate the spaces in order to create, or you cannot create order unless you have something separate from the disorder. That's the only way you're going to be able to do it. So you're going to exchange energy. Energy is going to be exchanged in and out of the cell so that you can create this order. Whether it be a plant cell or an animal cell, you have to be able, in order to organize things, you need a room. For instance, let's go to your room. If you have a room and you all have a bedroom, or at least you share one. My sister, her room was a complete wreck. If you couldn't walk in a room, her clothes were knee high on the floor. You had to push your way through it. You couldn't step or you'd fall. That's how many clothes were on the floor. I don't know if anybody else's room it was like that. Anyways, what do you do if your room is a mess like that?
Clean it.
How do you clean it? What do you do with all those clothes? You fold them, organize them, and do what? And put them where? In a drawer. What's a drawer? A compartment, a cell, a space. The way you organize things is you have to have spaces where you can create the order. It's the only way. Otherwise, its disorder will rule. So the universe is going towards entropy. Life exchanges energy for order. You need a cell to do that. At least as far as we can tell. There's probably other ways. If you read the case study, then you know that there's other thinkings of different ways you can do this. But as we look on planet Earth, all life on Earth uses these cells to exchange disorder for order. All right. So life is also made of matter. And matter is made of what we now know are the true elements. Elements are the smallest. Let me not say smallest. The purest form of something. The purest form of matter. So when you say I have the element gold, that means there's nothing in it but gold. There's no iron in it. There's no nickel in it. There's none of the other elements. There's 118 about, I don't know exactly anymore, elements. It changes all the time, or it changes periodically. 118 elements. You've seen the periodic table, I'm assuming. Here you have hydrogen. Over here you have helium. The first 4 rows are the rows that life is concerned about. The rest of this is just too heavy to worry about. The elements are too heavy. They're not very useful. Life is really worried about the first 4 rows of the periodic table. Let's take a look at an example, chemical reaction, and let's take a look at how these elements are put together. So let's take a look at water. When we look at water, an experiment that I would like to do, assuming we have time, is we can take water and run electricity in the water with some salt. We can add some salt just to help carry the electricity. But we could take water, and water we can break it down into hydrogen gas and hydrogen gas is H2 and oxygen gas. So let's be clear on what I'm talking about here. Water is an oxygen, an atom of oxygen, which is an element. Yeah. It says electricity. Electricity. Water is made of, is a molecule. It's sharing electrons. It's one atom of oxygen connected to another atom of hydrogen, and it's connected to two atoms of hydrogen. How do I know they're connected to two hydrogen atoms? H2. It says H2O. When you see H2O, it's because there's two hydrogens for every one oxygen. So that is water. Now I'm breaking that up on this side. This side, by the way, if you remember if you read the lab, you know this side of the reactants. And then what do the double-headed arrows mean? Why do I have a double-headed arrow? One in each direction. Yeah. That's right, because it's reversible. Excellent. And these are products. I know we didn't finish the lab, we didn't have time, and there was all kinds of... Say what? This class didn't start, but the other class started. Well, that's okay. I'm just saying, you read it. When I say I know we didn't finish it, I meant that as a ninth grade, as an honors biology group, all the classes together, not everybody finished it. Some people didn't start it, it's correct. So then here, hydrogen, on this side, hydrogen gas is two hydrogens bonded together. And oxygen gas, that's a plus. So there's two different things on this side. Oxygen gas is two oxygen atoms bonded together. And the G means gas. The G means gas. On this side, it would be an L for liquid. So liquid water can be broken up into hydrogen gas. Yes, I can zoom out some. And oxygen gas. All right. So what do you think, what do you see as a problem here? What problem do you see here? You should be taking notes. Wake him up. You should be taking notes, writing it down. Did you write what I just wrote down? Did you write down? See, I didn't write it, but did you write down that this means gas and that this means liquid? So you don't get confused when you see it in the book. All right. So what problem do you see between reactants and products? Do you see any problems? There's a big problem there. First law of thermodynamics says that you cannot create or destroy matter or energy. So what's wrong here? It's obvious, or I wouldn't ask you. So look on the screen and tell me, what problem do you see? There's a big problem. I'll wait another minute or two, let you see if you see it. The first law of thermodynamics says you cannot create matter or destroy it. That is the reactants on the left, that's what you started with. The products are on the right, that's what you end with. What is the problem? Is there a problem with what's on the screen right now? I'm telling you there is. What is it?
Ain't no hydrogen.
On which side?
On the product side.
These are hydrogen.
It's not on the reactant side?
There's hydrogen here on the reactants as well.
It changed form.
Say what?
It changed form.
It changed form. That's normal. That's called a chemical reaction. So we expect it to change. We went from one thing, water, to two things, hydrogen gas and oxygen gas. My question to you is, here's my question. We can't create matter, so what's the problem here? Look at it. What's the problem? We can't create it. We can only rearrange it.
It's because it's gas on one side.
No, it could be gas because it's just changing form. It was liquid, but now because these two are connected instead of these three are connected this way, this is a gas and this is a liquid. We'll talk more about why that is later. But my question is, count. What's the problem? Do you see? Let's lay it out. Let's just do it. On this side, I have hydrogen. I'm going to use a different color here. Hydrogen. I have two hydrogens. On this side, I have one oxygen. That's what I started with. I had two hydrogens on one. I started with two hydrogens and one oxygen. Everybody see that? One oxygen, two hydrogens. Now, on the product side, I also have hydrogen and oxygen. I do. On the product side, I have two hydrogens. That's okay. I started with two. I ended up with two. I'm good with that. Do you see the two there? It's actually H2. Do you see the lowercase 2? H2. But then oxygen, according to this, and by the way, oxygen gas is O2. According to this, oxygen, I have two. What's the problem? I only started with one. I can't create anything, so something's wrong. This is called an equation. This is my drawing of what's going on, so I have to improve my drawing. So what I have to do is I can't change this. I can't change the subscript. Those of you that are not paying attention are going to make this mistake on a quiz or a test. And when you make that mistake, know that I warned you not to make it. Do not change the subscript. The subscript, that's the lower little number, that tells you how many atoms of that element that's right before it is connected to the element that's right behind after it. So this says H2, so that tells you two hydrogens are connected to oxygen. If I wrote H2O2, that's not water anymore. That's hydrogen peroxide. I can't do that.
Well, you said don't change the subject.
Don't change the subscript.
What is that?
The little number. Don't change the little number. What we do is we add a coefficient. We add a coefficient. Now you have 4 hydrogens. Now you have 4 hydrogens on this side. Now you have 2 oxygens on this side. On this other side, you also have to add a coefficient here so that makes it 4 on this side. So that makes it 4 on that side. And now you have a balanced equation. That's how you balance equations. It's a lot of fun. We'll have to finish the rest of this tomorrow. I wish you guys would settle in quicker, though, and I wish the technology worked faster as well. Now what you have is you have a balanced chemical reaction. Because I'm adding 2 waters, just like you would add a cup of flour, and I'm getting 2 hydrogen gases and 1 oxygen gas. We call that a balanced equation. All right, so that's a chemical reaction. Now, can I go on? That's just an introduction. We'll come back to the chemistry of it later next week, where we're going to break down what are these covalent bonds, what makes a molecule, what makes an atom, how do you put them together, how do you rearrange them, and how do you know if a reaction is going to release energy or take energy in. In this case, in this direction, you have to put energy into the reaction. In the opposite direction, it releases energy. When you mix hydrogen and oxygen, you get an explosion. So next week we'll have miniature explosions happening in your test tubes, and you're going to make some water. Next week you'll pull water apart, and you'll put it back together again. But for next week, because we're dealing with explosions and fire, you're going to have your safety contract done. You're going to have your safety test done by next Thursday. Or Friday. Friday. Don't even look at me. Friday. As long as everybody, I'm just saying, if there's an individual in the room who hasn't, then we need to get that done before next Friday, so you can do the lab. It's a fun lab. I enjoy it every time. And it works out really well. I'm sure my assistants will enjoy doing it as well. All right. Now, when we look at, notice that I am doing Cornell notes for you. I'm trying to emphasize the reduction. A lot of you don't reduce. When I see reductions that I do in class, but I don't see things that you're reducing, that tells me you're not reviewing. That tells me you're not thinking about what we've talked about. You've been too busy in your side conversations. You've been too busy worrying about whatever it is you're worried about, and you're not taking care of your business in class. That's going to be the conversation I'm going to have with your parents if they come and ask me, my kid doesn't understand, my kid is not getting good grades. I'm going to say, look, they're not paying attention. They're offside, they're off task. Here's my evidence. What can we do about it? So, in any case, don't forget to put the date on every page and preferably your name all over the place, so in case we look at your notebook, when I look at your notebook again, I don't have a question. Excellent. So it's enough to know this, which is basically what's on this board. The elements of life. The elements of life. These are the elements that that all life on earth is made of. Carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. Carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. So tomorrow we'll try to answer that question that's at the bottom, the 4 basic molecules of life. By the way, anybody know the 4 basic molecules that are made of these elements? Anybody know the 4 basic molecules of life, the 4 categories?
Wait, what did that say?
Carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. The 4 categories are nucleic acids, DNA and RNA, lipids, which are fats, right? Proteins and carbohydrates. Those are the 4 categories of biomolecules that we'll talk about tomorrow. Don't worry, this is all recorded, and we'll start from here. Tomorrow's quiz will be what are the characteristics of life? Have a good one. Now, one of the things that we discussed, the question that you had to take home with you yesterday was, what 4 classes of molecules is life made of? That was your question. That was your homework. So now I'm going to pause for 3 minutes. You're going to write them down now without looking on the internet. I want you to write them down. If you did your homework, you have them written down, you may use your notebook. If you didn't, you have a 0. So write it down, put your name in the period at the top, and I'll collect them in 3 minutes. You have 3 minutes starting. Hey Siri, start a 3-minute timer. 3 minutes starting now.
Write it on a different piece of paper.
Howard, good job. Sana, good job. Real easy, right? Erica, Marie, good job. We know who did the work and who didn't. Easy question, but let's go ahead and delve into the details. Oh, hello. So, when we're talking about the 4 classes of biomolecules, there's 4 major biomolecules, and you've heard of them probably. Lipids, part of lipids are fats. So you've heard of fats, right?
Yeah.
But there are lipids also form steroids are also lipids. And by the way, there's something called fatty acids. All these are lipids, and there's different kinds. So make sure you're marking down what I'm saying, and making sure that you need to make index cards because you need to be able to use this information to identify in an experiment what kind of molecule you have. Yes. Yeah, grab it and take it and use it and then give it back before you leave. So lipids are one. Proteins are another. Look for it, dear. Look over there. Back there. There's 2 there. There's 2 there. There's 4 here. Proteins, which you've heard of before, I'm sure. Nucleic acids. And finally, the 4th is carbohydrates. I haven't introduced this word yet, but some of you may have heard it before. There's this thing called polymers. All of these here are polymers. They're made in a very special way. We're going to talk about it. This one over here doesn't make a polymer. Lipids are not polymers. But the other 3 are. And we'll talk about what that is. So lipids. Lipids are mainly, I'm going to use the word mostly, made of carbon and hydrogen. Carbon and hydrogen, the main component of lipids. There is some oxygen. There's some oxygen. And depending on the lipid, depending on the type of lipid, where it's used, etc., you will sometimes have that's right. You'll have phosphorus. Sometimes you'll have sulfur. And sometimes you'll have even nitrogen. All right, so sometimes especially the sulfur and the nitrogen, there are a lot of lipids in your body that have phosphorus. But mostly, again, the main characteristic of lipids, in fact, we call these things, another word for these, are hydrocarbons. Because they are mostly made of carbon and hydrogen. Mostly. Even the ones that have nitrogen, phosphorus, and sulfur, they're mostly carbon and hydrogen. And most of the lipids are going to be long chains of carbon and hydrogen. We'll get into details as we go on, but you need to know that much, that lipids are made mostly of carbon and hydrogen. Lipids are nonpolar. You don't know what that is yet, right? We'll talk about it. Nonpolar. So that's what you need to have on an index card. You need to start getting into your mind right now for the next test, for the first test you have, for the next quiz, certainly, for the next test, absolutely. you need to know these four biomolecules and these characteristics that I'm writing here. So if I were you, I would be writing these on index cards. Yeah. I would be writing this information on index cards. On one side, I would put lipids. On the other side, I would put all this information or split up into 2 cards, however you want to handle it. I've seen kids do it electronically on their phones. That's fine. My feeling, the research shows clearly if you write it down on cards with a pencil on paper, you're involving more parts of your brain and you tend to remember it more often. And if you write your own versus using someone else's, again, you tend to remember it more often. You don't know what nonpolar means yet, but everything else there you should understand. Is that okay? Is everybody okay with this so far? Any questions or concerns? Everybody understand what I wrote? It's neat enough? Okay. Let's go on to the next one, proteins. Proteins are, as I said, they form polymers. Again, you don't know what that is yet. You will in a minute. And they're made, which means, by the way, I should just put in parentheses, they have a monomer. No, I should say they have monomers. We'll talk about that in just a second. Proteins are made of carbon, hydrogen, nitrogen. All right, carbon, hydrogen, nitrogen, and oxygen. Sometimes, or some, you know what? I should just put sulfur as definitely one of them. But no, here's the thing about proteins. They have no phosphorus. This is going to be very key. It lets us understand a lot when we're trying to figure out what's what. They have no phosphorus. So notice they have all the other ones, but none of the proteins have phosphorus on it, in them. Proteins can be polar, nonpolar, ionic, acidic, basic. So they can be all kinds of different, they can have all kinds of different properties, and we'll talk about what allows them to do that very soon. Today, actually. And we're going to start with proteins, I think. Nucleic acids. So that's it for, by the way, there's the monomers. Again, we'll talk about what those are in a minute. There are 20 different monomers. That's why they can be any of those things, because there's 20 different ones. You need to think, say that again? Acidic, this is acidic. Ionic, acidic, or basic. So they can act like acids, they can act like bases, they can be nonpolar, they can be polar, they can be small or large, they can be any kind of shape. They form all kinds of shapes. Proteins are the structural. When it comes to function, I guess I should mention function for lipids as well. When it comes to function, lipids are structural. In other words, they help build your body, your cells. Your cells' one of the main components of your cell are lipids. Lipids are long-term storage of energy. when a plant wants to store energy a long time, it puts it into oils. Oils are fats. Oils are lipids. All right. So sunflower oil, corn oil, we use it to fry food. That is the plant's long-term storage form. We have fat on our body. This is our fat. This is our lipid. This is our long-term storage of energy. It can also help insulate. When you see a walrus and a whale, they have something called blubber. It's a type of fat that really is dense and allows them to keep warm in the cold oceans. So fats can do a really good job at insulating your body. She explains why larger people like myself are outside and the cold. You'll see some of us walk around no problem in the cold and you're freezing, shivering, turning blue, and we're just looking at you like, why are you cold? And then in the heat, in the hot, you guys are running around in the heat. You guys are running around with hoodies, and I'm sweating in the shade, wondering what the heck is wrong with you. My body fat insulates me a lot more. Fat is less dense than muscle. So it's easier to float. It's easier to float in water. Thin people find it more difficult to float in water. Dense, yeah, dense. You know, you remember density from middle school? Like more stuff in the same space. Okay, so that's pretty much for fat. Proteins, they're more dense as a whole. Like muscle is made of protein, for instance. A lot of your body is made of proteins. Proteins are the functional unit. They do involve some structure. They're involved in function. Proteins do most of the work in your body. They do most of the chemical work in your body. Muscle, as I said, is made of protein, so you move because of protein. Enzymes are made of protein, so you do what you do because of proteins. Your hair is made of protein. Your skin is made of protein. Bones are not made of protein, no. It's a different substance. But proteins make up a lot of your, well, I shouldn't say that. Bones have protein in them, but not. Do you have a tardy pass? Did you sign in? Fill out the tardy slip? Could you sign in to the, why are you staring at me? There's a little table there, and it says tardy pass, remember? You've been tardy enough, you should know. All right, nucleic acids. There's two types, right? Who knows what those two types are? I know you know. That's right. DNA and RNA. These are also polymers, so they have monomers. They have DNA has 4, and they are A, T, G, and C. Adenosine, anyways, you'll see. 4 different monomers. That's it. You'll need to know these very well. You'll have to read genetic codes and distinguish between 2 different genetic codes. So you might as well start getting used to knowing what these are. RNA has 4 monomers as well. It's A, U, G, and C. It's the same 4 except for the T is a U in RNA. They have, these are not elements. Does everybody understand that? These are not elements. A, T, G, and C are symbols that represent monomers. Am I being clear? So don't get confused. Nucleic acids, both DNA and RNA, are made of carbon, hydrogen, oxygen, phosphorus. But they have no sulfur. That's going to be key. Again, when you're trying to solve problems, when you get problems on quizzes and tests, the fact that there's no sulfur in DNA can be very useful to know. And the fact that there's no phosphorus in proteins is good to know. Carbohydrates. What is the function of DNA and RNA? DNA is where you keep your inheritable, something that you can inherit, information. That's where you send your information from one generation to the next, is in DNA. RNA has basically 4 functions. It actually does a lot more in DNA. It can tRNA, there's 4 kinds, transfers amino acids. You don't know what that is yet, you'll learn very soon. RNA, rRNA makes up ribosomes. Those of you that did cells in middle school probably remember ribosomes as being an organelle. rRNA make ribosomes. Ribosomes are made of RNA. So in fact, RNA can act as an enzyme. Again, you don't know what that is yet. I'm just giving you information. It'll all make sense, I think, as we move along. Okay? So RNA can act as an enzyme. There's another form called mRNA. It acts as a messenger. So when you see these, and I use the words, hopefully they won't be new to you. It'll make a little more sense. If you, again, put them on index cards, know what they are. These 4 kinds: tRNA, rRNA, mRNA, and the general RNA can act as an enzyme. So 4 different purposes of RNA. DNA only has one purpose, and that's to keep your information safe. Don't you think that you want to make sure that the information that makes you who you are is kept safe in a vault?
Yes.
Right? That's precious. In the cell, the most precious thing you have is your DNA. Everything else is about getting that DNA from one cell to the next cell. Most of this is extremely important. All right. Carbohydrates, your favorite. Your absolute favorite. They are sugars. All right. They have their single form, which we call them sugars. Another word for sugar, before I go on, is saccharide. Has anybody ever heard of a saccharide? Yeah. You might have heard of a word called saccharin. It's a brand name for a synthetic form of sugar. So saccharide is the word that we use for sugars. So anything that has only one, that does not a polymer, only has one unit. So if I had a bunch of single units, we would call it a monosaccharide. An example of a monosaccharide would be glucose. Glucose is a single unit. Looks like this. That's it. Single unit monosaccharide. There's another monosaccharide that you should know. So I think everybody's heard of glucose, right? By the way. Okay. There's another one you probably have heard of is fructose.
Yeah. What's that?
Where do you think fructose comes from?
It's glucose.
Think.
Is it fruit?
Fruit. Yeah, fructose comes from fruit. Very hard, much harder for you to digest than glucose, by the way. And where you find a lot of fructose is in soda. They use high fructose corn syrup. What that means is there's a lot of it. High concentration of fructose. That's really not good for you, just so you know. It does damage to your liver. It obviously stimulates the whole issue of diabetes. So be careful with drinking those sugary sodas. It's not just that they're full of sugar, it's that they're also full of fructose. And that can cause a problem. There's another one you probably have not heard of that's going to be very important to you is ribose. Glucose and fructose have 6 carbons. Ribose has 5 carbons. This is going to be important, so remember that. If I'm not putting information down, you don't really absolutely have to know it. You can know more. There's a lot more to learn, but I'm giving you the bare bones that you should put on index cards and make sure you know. You've heard of another one that I know a lot of you have heard of is lactose. You've heard of lactose? Where is lactose found? Milk. Why do we call it lactose? You're right, it's in dairy, milk and such. Why do we call it lactose? Because, yes, in part, it doesn't always come from a cow. It's also found in human milk and in any mammal milk. Because it's lactated. What does lactated mean? It comes from a breast, right? Babies drink it. So baby drinks lactose. Babies are born, when babies are born, I say babies, I mean a baby cow, a baby goat, any baby mammal. Baby whales drink milk. Did you know that?
No.
Whales, they go underwater, they suckle at the mom's teat. We call it a teat. Not the other horrible word. That other word should not be used for a human. Is that clear? The word is teat. I didn't say any other word. Let's not bring that up. That's the official word for animal breast. Are we clear? Yeah. All right. So baby whales do suckle at a mother's teat, and mother does lactate. They produce milk for the baby whale. How much milk does a whale produce? A lot. I have to imagine gallons, right? I've always wondered would it save money to go and harvest milk from a whale instead of a cow, right? I don't know. I've always wondered about that. Anyways, lactose, ribose, fructose, glucose, really big deals. By the way, the reason that most adults really should not be we're not made to drink lactose after we're grown.
That's why a lot of people lactose
That's why most people can't really drink a lot of lactose as you're an adult. It's not made for us. It's made for what? Babies. Babies have enzymes that help them digest it. As you get older, you turn those enzymes off. You turn those genes off because you're grown. Now, we kind of cheat a little bit. We can turn them back on, but it takes a while to get them turned back on. And some people are born with a deficiency in that. So they have to take lactate, which is the enzyme that babies have that allow them to digest it. You don't know what an enzyme is yet, so just bear with me, okay? I will explain it to you. But just know that that's why people get upset stomachs, milk farts, even to the point of diarrhea if they drink milk. Most of you never drank whole milk. Anybody ever here drink whole milk?
No. I drink
All right. Most people drink 2%, some people 1%, but very few people drink whole milk. Almost no one has drank milk right from the cow.
Why would you do that?
It's very dense, has a lot of lactose in it. It's fresh milk, you can drink it. That's where all milk comes from, or at least the milk we drink. Mr. Mendoza's room, may I help you?
You gonna be on the toilet all day.
Mm-hmm. Okay. Yeah. I appreciate it, yeah. I appreciate it. But may I tell you, though, that we got an email from Maria, and she would like to make sure that we have something in our presentation to the parents. Because I know you're she says she might show up. I don't know what that means, but she said it to everybody, not just us. So I thought maybe I'd make a poster and to put out in the front of the office. Yeah, I finished the letter. I sent it to you, but I don't know if you want to change anything. And all right, and maybe part of the presentation, in case she shows up with somebody and she knows that we mentioned it during. All right, I'm sorry to do that, but I was like, if I don't tell you, it's going to be a mess. All right, bye bye. All right, lactose. So these are the 4 sugars. These are 4 monosaccharides. There's a lot more. There's galactose. There are babies that are born with being galactose intolerant. They used to die, actually. It's called maple syrup disease. Do you know why?
Oh, because
Now think about being a doctor in 1700, 1600. You didn't have a lot of labs. You didn't have a lot of tests. You did not have a lot of chemical tests. But you have a baby who's sick. What did doctors do? What in ancient Greece, in the Middle East, in China, what did doctors do? They looked at poop, right? To see, and it studied poop because that came out of the body, right? It didn't actually, poop, by the way, does not actually come out of your body. It's just what's left over from what you didn't digest, what you didn't take into your body. Poop is what's left. That stuff that comes out of into the toilet from your butt, that was never in you, all right? That's what's left over from the stuff that you couldn't take into your body. What comes out of your body is called pee. Does that make sense? So urine comes out of your body. So doctors today, we still do it today, by the way. We still look at poop. They'll take a poop sample, they'll smear it, they'll study it under a microscope. Back in the day, they didn't have microscopes, right? They didn't know what bacteria was, but they could tell if somebody was sick by looking at the poop, at least to some extent. And if it was the color, the smell, the texture, what was in it, right? All tells you a lot of information. What else could doctors do? They could smell it, they could feel it. Of course, they probably didn't use gloves back in the day, but they wash their hands. Today we have gloves. They didn't have a lot of gloves, like rubber gloves. Do you understand that? Like rubber gloves is it's a modern privilege. So they tested the poop. What else would they test? How would you test the pee? You look at what? Color. Okay, what else? Smell. You know when you eat asparagus, it smells like asparagus, right? You ever eat asparagus?
I
Oh.
love
Amazing.
asparagus.
If you ever eat asparagus, you can smell and go pee afterwards. You'll smell the asparagus. So you can actually smell. You can smell a lot. You can learn a lot from smelling pee. Guess what else they did? Not drank it, but they did taste it. They would dip their finger in the pee and taste it. Again, they didn't know anything about viruses. They didn't know anything about bacteria, correct? Hey, you think it's funny, but it actually saves lives. It saves lives. If the pee is sweet, they knew they had what they would call wasting syndrome, which today we call diabetes. So if the urine was sweet, they knew the person had a problem. Most people with diabetes back then died. So knowing it early, being able to diagnose it early, say, look, you got to eat less sugar, that helped, but they still probably died early. Because remember, they didn't have insulin. They didn't even know what that was. So then, babies would be born very sick. And when you tasted their urine, it tasted like maple syrup. That's because they could not digest a sugar that's found in milk. It's called galactose. Galactose is something, and lactose is actually not a monosaccharide. Lactose is, all right, so anyways, lactose is a bunch of different other sugars, but when you look at these sugars, what you're going to find is that we can connect them in a lot of different ways. These that we're talking about here are really about energy source. They're about energy, short term. If you eat too much sugar, it doesn't stay as sugar. It gets turned into a polymer or it gets turned into fat. If you eat too much sugar, it does not stay sugar. You can't. It'll kill you. Yeah.
What is that first thing you said?
What? Polymer? Oh, I'm going to get into that. It's going to take a minute for me to describe. But a polymer is, have you ever built a Lego set? Anybody here built or seen a Lego set get built? Well, a model of a Lego, if I built you from Lego blocks, the model, the statue would be the polymer. Each individual block would be a monomer. All right, so polymer is when you take a bunch of units and stick them together in a certain way and you get something big. Okay? So that's what it is in short. I'm going to go into detail on it. But probably not today because we're running out of time. So short term, the monomers and lactose, they're used as, and actually I should say disaccharides as well. They're mainly energy. For instance, table sugar is a disaccharide. It's two monosaccharides put together. So when you eat table sugar, it's not glucose. Glucose is only one of the two. You create plants or we refine it from sugar cane or beets, and you can make it's a disaccharide, two monomers put together. But you can form polymers, a bunch of them put together. And when you form polymers, they can do long-term, medium-term, to long-term storage. Of energy. The examples here would be in humans, in mammals, we have something called glycogen. Glycogen is stored in the liver in humans, and it's just stored temporarily. If you get low blood sugar, your body releases the sugar from where? From the liver, from glycogen. So you take a bunch of glucoses, and then you take a bunch of them and you stick them together. And as you stick them together, they form these big long chains, and you store them in the liver. Does that make sense? They form these long chains, and you can just keep them in the liver. They're really long. Your liver stores them for when you have low blood sugar. You can quickly release sugar into your blood so that you don't pass out. That's important. That's in animals. So glycogen, this happens in animals. Glycogen is not found in plants. Plants use something else. They put it in their roots, in potatoes, in beets, in yams. Are beets? Yeah, beets are roots as well. What they do, it's actually not beets, but in yams and in potatoes, they use something called starch. You've heard of starch? That's just more of this medium or to long-term storage. We steal their starch. We steal their sugar by digging them out of the ground and frying them in a pan. We call them French fries. But all we're doing is frying sugar. French fries are sugar. Huh?
Like starch, it could bring up your iron.
It can bring up what?
Your iron.
The potato skin has iron in it, but the starch is just sugar. Starch is just sugar, and anything that's starchy is just sugar. Batata, I don't know if anybody is familiar what that is, but it's a root. Candy yams, they're all sugar, starch. But they don't have to be, you can take the same sugar that you eat, you can put it together in a different way. Remember we said Legos, you can put them together in different ways, right? You can put them together in a certain way, and you get something called cellulose. This is very important. You're writing on it right now. You're writing on sugar. That paper is made of sugar. That stool you're sitting on, the wooden stools you're sitting on, they're made of sugar. Wood is made of sugar. The sugar is called cellulose. Almost nothing can break it down. There's only one. Even trees, when they make this, they can't eat it. They can't break it. They can't break it up. Plants, everything that the structure of a plant is made of cellulose, made of sugar. But they can't break it down because they put it together in a way that it can't be broken down by water. The only thing that can break it down is a tiny bacteria that has a special enzyme that can break it down. So it's made of sugar, but almost nobody can eat it. There's only some fungi, fungus, and some bacteria that can eat it. Termites cannot eat wood. But in their guts, they have a bacteria that helps them eat it. They'll chop it up, but then in their guts, they have a bacteria that helps break down the wood into sugar. That's why they eat the wood, because it's made of sugar. So consider that sugar is not always something to eat. So cellulose is structural. And that's a short breakdown of the biomolecules. Tomorrow it looks like we'll get into what polymers and monomers are. It's a little messy. Sorry about that. So tomorrow we'll get into what polymers and monomers are. Okay? So the only change is, as you revise your notes, as I keep telling you, one of the things you need to do as you're taking notes and you're becoming a professional, you got to go back and revise your notes and correct any errors or add some stuff the way you're supposed to be doing with your half of your assignment, correct? Am I correct? In half of your assignment is for you to take your summary, your chart, and add the stuff that's not on that chart from these notes, right? So everybody understands that, good, because I don't want anybody saying they didn't get it. And so here we have, why is it doing that? That's strange. Record from here. So one of the things you have to correct is that RNA has, all these were correct, except you have to add a big one, which is nitrogen. That's why they're called nucleic. One of the reasons they have nitrogen in them. All right. Think of nucleic NN. Okay. Think about this being an N and nucleic having an N. If you're having trouble remembering that, let me do a different color here. Nucleic has an N and nitrogen is an N. All right. So they have phosphorus and nitrogen in them. They're called nitrogenous bases, but we'll talk about that later. Then the other thing you want to make sure you write down and correct, not really correct, but that lactose is a disaccharide, which means that it is made of two sugars. All right, di means two, and saccharide, as I said earlier, means sugar. All right, so it's a disaccharide. The lactose is made of galactose and glucose. All right, so you're going to take these two blocks. I'm going to put hexagons. You're going to take these two blocks, two monomers, and you're going to connect them to form a disaccharide where one's glucose and one's galactose. Those two together make this thing called lactose. And that's the sugar in milk and in cheese. Okay? So it's called lactose. There's another sugar called sucrose. And that sugar is connecting glucose to fructose. So those two sugars come together in a disaccharide. Those are two examples of disaccharides. So table sugar has glucose and fructose. Glucose and fructose. So when you're sprinkling sugar on your stuff, you're putting glucose and fructose on there. Lactose is glucose and galactose. So there, I know it seems like it's a small thing, but there are babies born that cannot digest galactose, and if they keep giving them mother's milk, right, they die. So the trick to saving these children, at least in the earlier years, is to not give them galactose as a sugar. You can give them glucose or fructose, but you can't give them galactose. Yes, question?
So what does it mean when somebody's lactose intolerant?
That's a really good question. So these are connected. In order to digest these, in order to absorb them and digest them properly, you have to break them apart. These are connected. Do you agree? We connected these. In order to eat this and really use the energy that's in sugar, you have to break these apart. And we're going to look at that in just a minute. In order to break them apart, you need something called an enzyme. An enzyme is something that allows, normally sugar is very stable. For instance, you know that there are wood houses that have been around for hundreds of years. Do you agree?
Yeah.
I would even argue there may be wood that's out. There's probably wood out there that's 1,000 years old. So if you have this 1,000-year-old wood that's made of sugar, you agree sugar can last a long time. Yes?
Yeah.
So it's very stable. If you waited for this to break apart by itself, you'd be waiting a very long time. And you need sugar now. When you're low energy, you need sugar immediately. When you're a baby growing, you need sugar quick. So you have to find a way to break this apart quickly. Well, what you use is an enzyme. So in comes, I'm just going to draw a very simple structure just because it makes life easier. So you have something that looks like that. Well, the enzyme is going to look something like something. It's going to have a shape that lets this fit in like a key. Say that again. It's just a globule. It's an amorphous hard. Well, it's not amorphous, but these enzymes are protein chains that don't really have. They don't really have a shape like a bird, like you could identify quickly like a bird or a human. They're odd shapes, but the enzyme will this thing here. Maybe that's a better picture. This lactose will fit in the enzyme. And what do you think the enzyme is going to do? I like eating it. Break it is a good idea. Yeah. It's gonna speed up the reaction. If it was happening on its own, this would not break apart within the lifetime of the baby. It would just stay together. But because the baby has this enzyme that the baby made, that you make, okay, this enzyme that you make, then this chemical reaction can move forward. And we'll talk more about what that is in just a little bit after we finish describing what these biomolecules are. So what happens is at the end, you have this same enzyme, okay? In the end of that arrow is a chemical reaction. You all agree? In the end, you have this. The enzyme never changes. The enzyme does not change. What changes is the sugar. And so because this doesn't just happen for sugar, it also happens for other things. We have to give the reactant here, the thing that's changing, we have to give it a name, and we give that thing the name substrate. All right, substrate. So the substrate is the thing the enzyme acts on. So you start with glucose connected to galactose, and you end up with a glucose and a galactose. Do you understand that? Does that make sense? That's how the baby breaks it apart. Yes? That's how you break it apart. When you drink lactose, if you digest it well, this is answering your question. If you digest it well, right, you have this enzyme and you're able to break it apart and then you can use it, no problem. You have no problem drinking milk. You have no problem eating cheese. Is that clear? But if you're lactose intolerant, what happens is that you can't make this enzyme. And we'll talk about how we make enzymes soon. So you cannot make this enzyme. And because you can't make this enzyme, here you sit with just glucose and galactose that you're drinking this milk, you're eating this cheese, but you can't digest it. Well, what else do you have in your intestines? Do you know what other stuff you have in your intestines? Acid. Well, you have acids in your stomach, so let's go ahead and look at the digestive system. It's something important you should have some idea on. You can make fun of my drawings if you want, but you do it at your own peril. You know what that means? You do it at your own. You're risking your. I'm kidding. I was just making a joke, but you don't understand it. Okay, fine. So this is your mouth. All right. So things go in. It's a horrible drawing that's not really meant to be a drawing. It's called a sketch. I'm just trying to get some... I shouldn't even draw it on that side. It's a sketch meant to make a point. It is not meant to be an artistic drawing or representation of any real thing, all right? It literally is meant to be exactly what it is, okay? And I think because you're laughing, you have some idea what it is, all right? I should maybe draw something more similar to the shape of a stomach, I suppose.
Does your letterhead break it down?
It does, but in another... Let me undo that. It's hard to draw this. And represent it well. You know what's coming out here, right? That's right. You know what went in here, right? I think food went in, right? You agree? What do I mean by food? I mean protein, lipids, I mean carbohydrates, I mean nucleic acids, and then minerals. Minerals and things you call minerals are rocks, sodium, potassium, calcium, that kind of stuff. That goes into the mouth. I want you to understand something. Your body's here. Your brain's up here, your eyes, your blood is over here. Okay, that's on this side of... You surround this tube that's going around here. Do you see that? This is a 2-dimension, but you have to imagine this is 3-dimensional. Are you understanding when I say to you that when you eat something, that thing is not in your body? When you poop, that is not in your body. The poop was never in your body. Do you understand that? Because anything you eat is in this tube here. Anything you eat is in this tube. That's how when you eat poison, they're able to get it out of you before it kills you. Why? Because you vomit it, they make you vomit. And since it was never in your body, it was just in your stomach. It was inside the stomach, which is what? A space, a cavity that your body surrounds. Your body surrounds your intestines. Your body surrounds your colon. Your body surrounds your esophagus, right? That there is outside, right? And it stays outside all the way through. So when you poop, that stuff that comes out of you was never in your body. It's the stuff that was never in your body. Is that clear? Yes or no?
Yes.
Okay. You need help digesting. You need help breaking stuff down. There's stuff that you cannot break down. There's stuff that you can't really take advantage of. If you are lactose intolerant, you don't have this enzyme. You have this really fantastic group of living things called bacteria. They're very, very helpful. We have helpful bacteria. There's harmful bacteria as well, okay? Somebody said acid. You're right. There is acid. That acid is here in your stomach, okay? Is that too messy for you? Awesome. Acid. All right, so that's acids in your stomach. There's no acid anywhere else, at least no strong acid, anywhere else in the system. You have special cells that help protect you. You put acid in your stomach, that's okay because it's not in your body. If that acid ever got in your body, that would cause you damage. This is in your body. Your liver, your kidneys, your blood, your brain, your eyes, they're in your body. Is that clear? This is still outside. Then in the intestines, you have these really billions and billions and billions of helpful bacteria. How did you get the bacteria in your intestines? It wasn't in your mom's womb. Your mom protected you against bacteria. How did you get the bacteria in your body when you were a baby? No. What do babies do? Put everything in their mouth. Do you ever notice that? They go around licking everything. They'll lick the wall, they'll lick the floor, the spoon. Do you ever not notice that? It's not that they're hungry. It's on purpose. It's not an accident. Babies are programmed to do that. As soon as they're born, the first thing they start to gather bacteria to help them digest. You ever seen baby poop?
Yes, solid.
Do you think, and smelled it, do you think it looks like your poop at all? Not even close. Baby poop it's hard for them to digest because they don't have a good collection of bacteria. So they spend a long time going around collecting bacteria into their intestines. And they fill their intestines with bacteria that helps them digest. Your intestines are filled with bacteria, hopefully good bacteria. Now, if you don't make this enzyme, that means you can't break it up. That means this intestine is full of this lactose. What do you think that bacteria is going to do? They eat it. They're going to eat it. They do have the enzyme. They have these bacteria in the intestines. They have the enzyme to break this up. And so they eat sugar. When they eat sugar, they make gas. When they break this down, they break it down just like you do with an enzyme. And they make glucose and they make galactose. And they eat the galactose, they eat the glucose, everything's great. You're not getting any of that sugar. The bacteria is getting it. They're multiplying. They're making gas. What does that do to you? What does the gas do to you? Well, not burp, because it's not coming out of your mouth. So your gas builds up in your intestines. You get cramps in your stomach. Your intestines get painful. Do you ever have bad gas? Without farting? Before you fart? Did you ever have bad gas? It hurts. It feels like cramp, like your whole intestines cramping up. In fact, when you fart, you're happy. Because it's like, oh, thank God, it's out. It's painful. And someone who's lactose intolerant, what happens is they get gas in their intestines. They get water rushing in because the bacteria are multiplying. So they get diarrhea. They get really bad cramps in their abdomen. It's not great. So they shouldn't drink milk. And most adults cannot handle a lot of glucose because we do not make this enzyme. We stop making it after we're babies. As humans, after we're done being, most mammals, after they're done being babies, we stop making this enzyme. That's why we allow fungi, a fungus, to eat the milk and make cheese for us, right? We love cheese. Even somewhat, if you're really lactose intolerant, you can't even eat cheese. But most of us can eat cheese. Very few of us can drink whole milk. Most of us, if we drank whole milk, we would get sick. Our stomach would hurt. But most of us drink 1% or 2% milk, correct? When you're eating cheese, you don't have a problem with it. Why? Because you allowed the fungus to break up some of that lactose, a lot of that lactose, and make this thing called cheese with different flavors. That's because of the fungus, which is another living organism, not a bacteria, helps break down the food for us. So we often use other creatures that help break things down for us. Termites. You heard of termites, right? What do they eat? Wood. Well, they don't eat wood. First of all, wood is cellulose. And cellulose is, just like we see here, is a chain of carbohydrates, a chain of sugars, all in a row. And I'm just sketching it out real quick. All connected in a row, in a big chain. It's connected in a way that very few living things can break up. We can't eat it. We could eat wood all day long. We couldn't break it down. We couldn't break it up. It would come out of us all wet and slimy and disgusting if it didn't block up our intestines and kill us. So eating wood is not a good idea for us. Termites have bacteria in their intestines that they live with. They don't really have intestines. They have little cavities in there because they're insects. But they chew the wood, like someone said, they chew it. They put it in their digestive system, and those enzymes help break up the cellulose. So they don't digest cellulose either. They break cellulose up into sugar. They eat the sugar that chair is made of, but the bacteria helps them digest it. So digestion has to do with breaking up these polymers, is the point. Did that help you understand what lactose intolerance means? So if you're not tolerant of lactose, first of all, almost no human adult should be tolerant. We should all be tolerant, but not easily digested. Babies are not only tolerant, they love it. They eat it all day long because they have the enzyme. Is that making sense to all of you? Since you all say yes, I'll put it on the next quiz, okay? So you'll be able to describe it to me, correct? All right. So make sure you can. All right. So someone asked, what's the difference between glucose and galactose? Let's take a look at that. Glucose looks like this. Galactose looks like this. There are babies born that cannot digest this. They cannot break this up. They cannot break it up. And because they can't break it up, if they keep drinking milk, they die. It's a really, really bad, it's called maple syrup urine disease. The ancient physicians in Persia and in Greece, they identified the disease, and the way they did it, not drinking it, they tasted the urine. They would taste some of the urine, it would taste like maple syrup, and they knew that the baby wasn't digesting the sugar right. So they would prescribe, don't drink milk, because that'll help save your baby. Because milk has galactose in it. Now, they didn't know why. Because they didn't understand what molecules were. They didn't know that lactose had glucose and galactose, two of them put together. These two together, they're not connected here. You see they're not connected. These two connected together would form something called lactose, the sugar in milk. Well, what's the difference between them? They look very similar, don't they? The only difference is the OH here is pointing up, the OH down here is pointing down. That small difference means the world to the babies that are born that can't digest galactose. They cannot break this apart. You might ask, well, that seems kind of petty. Don't you think? Seems kind of small. But let me ask you, if you had a key that looked like this, it worked, and somebody else had a key that looked like this, would it work in the lock? So it's a small difference, right? They have two little prongs. This one's pointing down, this one's pointing up. But this would work and this wouldn't work. Well, it's the same thing here. The reason that babies that have this disease can eat this and not this is they don't have the key that helps break this apart. Does that make sense to you? This doesn't fit in the lock. This key has a different shape. It doesn't fit in the lock, so it doesn't work. So that's how enzymes, that is how the basic idea behind enzymes. They have to fit. Most of you're literally like the Lego blocks that we discussed yesterday in Minecraft. You build yourself up with shapes. They're not shapes like triangles and circles. They're really crazy shapes. They're really complicated, crazy shapes. They're not, I wouldn't even begin to describe them. But they fit. One fits into the other. One either fits or it doesn't fit. If it fits, then the chemical reaction moves on. If it doesn't fit, then the chemical reaction stops. Now that's a simple explanation. We'll get into more detail in just in a little bit. Are we all okay with that so far? Nobody has any questions? Great question, by the way. Thank you for that. Let's move on. Let's try to... All right, so here you have this document. You see it has the drawing. You see it has the glucose, the carbohydrates drawn. This is glucose, oxygen up, oxygen, By the way, there are some small groups you should start to learn what they are. So I'm going to draw these out over here for you. You should make sure you can draw them. So there's this thing called OH. That is known as a hydroxide. Hydroxide. Anytime you see an OH together, that's a hydroxide. Now, where would you see that in this molecule? You see it here. There's an OH. There's a hydroxide. There's a hydroxide. There's a hydroxide. There. Do you see that? Does everybody see that? Say that again? So for some reason, you didn't get I'm sure there's copies somewhere. I just don't see them anywhere. Well, whatever. I don't care. Hydroxide. Hydroxide is a functional group. So these, when we look at these molecules, we try to understand how they work. So if you're going to be a physician, who wants to be a nurse in this room? A nurse? Who wants to be a doctor? Okay. Who wants to get anything in the medical field, biology field, veterinarian? Okay. So you need to know this stuff. Eventually, you'll need to know it cold. So start to learn it now. This is called a functional group. No, that's a good question. We find these in carbohydrates, yes, but you'll find this functional group in all biomolecules. I'll show you in just a minute. So one functional group is something called hydroxide. Another functional group is something called carboxylic acid, also known as, or carboxyl group. So C, it looks like this. I shouldn't use these dashes, but I'm going to go ahead and use it. So you'll see a dash connected to carbon. That'll be double bonded to an oxygen, and there'll be a single bond to a hydroxyl group. This is known as by another name, and that name is carboxyl. So this whole thing together, hydroxide with a C double bond O, is called a carboxyl group. All right. Yes, good question. Yes. She asked, are there functional groups in all molecules? In all biomolecules, yes. In all the molecules of life, there are functional groups. Excellent question. Which functional groups are where help define how that biomolecule works. So this is a carboxyl group. There's another group called an amino group. And that's this nitrogen connected to two hydrogens. It's called an amino group. All right. And then there's the last big one. There's many of these, by the way, but these are like the four big ones you're going to see. There's a phosphorus with 4 oxygens surrounding it, and we call this a phosphate. Now these are all chemical bonds. These are small little units of atoms sharing electrons. We call these functional groups. Question?
Why on some of them you only drawing 2 lines and the other ones you're drawing 1?
It's a really wow. You're on it today. Thank you. I'm not joking. That's exactly it. That is chemistry, and I'm going to get into it probably on Monday. But suffice to say that each atom can make a different number of bonds. So, for instance, carbon can make 4 bonds, can share 4 separate electrons. Yeah. And I think someone just left. If the pass is back, yes. Oxygen. Is it an emergency? Oh my God! Like, you guys can't be having this many. Everyone having an emergency at the same time. Seems so odd to me. You're using this one time and bringing it back to me. Hurry up. Oxygen makes 2 bonds. Those are the big ones you have to know. Hydrogen only makes 1 bond. Nitrogen can make as many as, let's just say it makes 3 bonds. This changes depending on conditions. Phosphorus can make 4 bonds or 5. Carbon makes 5 bonds, all right? 5 bonds. So if it doesn't have a bond, as I said, let me get to that on Monday, okay? Because to understand this, why this happens, you have to have a periodic table in front of you. You have to have a drawing of an atom. But I think some of you have heard of valence shell electrons in middle school. All right, you heard of bonds? Yeah. It's a good question. All right, so I see we're not going to let this go, so let's go ahead and... All right, so let's take a look at a periodic table. I know you can't, so let me zoom in. Is that better? I know it's a little blurry, I apologize. So periodic table tells you a lot. The group number here tells you what the number of valence shell electrons an atom has. So I'm just going to keep everything very simple since this is not chemistry. An atom has a number of protons. On this periodic table, on every periodic table, you have the number of protons. They're here in this number. You can look at, it's called the atomic number. The atomic number. The atomic number is the number of, I don't know why I do, why would I leave out a B? I have no idea. The atomic number is the number of protons. Is a proton negative or positive? Positive, that's right. Somebody remembers their middle school science. A proton is positive. A neutron is neutral. Zero charge. An electron is negative. There are two rules to chemical bonding. Two rules, if you remember them, it'll help you the rest of your life. Every atom in the universe wants to do these two things. They want to have, number one, a full outer shell. I'll explain to you what shell means in a minute. That's the first rule that all atoms want to do. They want to fill their outer shell. And number two, they want to be neutral. Nothing likes to have a charge. Everything wants to be neutral. Did you make those copies, darling? Thank you. Could you open the door? Somebody, I don't know why they all had to go to the bathroom at the same time. I don't know why. But full outer shell, and they're neutral. So when we look at these at the top, at these column numbers, you see these column numbers here, group 1, group 2, group 3, group 13, 14, 15, 16, 17, 18. Do you see that? Yeah. All right. So again, I'm keeping this very simple. It's gonna get more complicated when you get to chemistry next year. But this group number tells you the valence electron number. What that means is how many electrons on the outside, the outer shell. What do they want? They want a full outer shell, and this tells you how many electrons they have in their outer shell. Is that understood so far to everybody? So you got an atom, and I will repeat it, and thank you for asking. You have an atom that has a number of protons. Let me give you an example. You have a hydrogen atom, has 1 proton. Every atom has a certain number of shells. The shell that's closest to them the first shell can only support 2 electrons. Max has a max of 2 electrons. Everybody okay with that? Notice me revising the notes. Do you see that? Okay. So hydrogen only has 1 proton. How many electrons do you think it's going to be able to pull in? This shell can hold 2, but how many electrons can this 1 proton pull? What is the rule? They all want to be what? Here's 1 plus. How do you become neutral if you have a positive charge? 1 electron. 1 negative would make it neutral. So 1 electron is all it can pull in. Now this thing is, so you can check off, it's neutral. Neutral, check, no problem. But does it have a full outer shell? It does? How many can it fill? What is the filled outer shell for this atom? 2. How many does it have? Is it full? It is not full. So this has 1 proton. Who can tell me what element it is? It has only 1 proton. Hydrogen, correct. So I know this is hydrogen because it has 1 proton. That's it. There's a lot of ways hydrogen can get that extra. It needs how many electrons to fill its outer shell? It needs 2. It needs 2. Now, it's very rare that something like hydrogen, and again, I don't want to get into the details. There's all kinds of math and words that we could start using. This is also neutral, by the way, but it's not full. They're both hydrogen because they only have one proton. There's all kinds of ways that we would be able to talk about how you can get there, but let's go ahead and add this another element here. What if I drew something that looked like this? So 8 protons, you can count them, and you should draw them. This is a really big atom, and it has 8 neutrons. I'm just going to draw the neutrons as green. All right, so it also has one. I shouldn't have drawn it as green because I need to use the same thing for the orbital. So, okay, so this is an atom. It has a shell as well. Oh, this pencil just died. The pencil just died, so I forgot to charge it last night. So please forgive me. I'm gonna have to write with my finger. I don't know why it's, is this reversed? So again, I apologize. I'm going to have to draw with my finger now. So there's an orbital here. Are you kidding me? Come on. Sometimes he tried to spit on it because he couldn't beat me up. So he spit on it. Guess what I did? I went, Gah. He'd look at me, go like, he'd go. It's like, yeah, I'm getting mine. Period. Pizza is my ice cream. I love pizza. Oh yeah. So there's a partial charge. The delta means a partial charge. So when you unplug something quickly or plug it in quickly and there's a spark, that's a real charge, right? A lightning is really charged. You agree with that? That's kind of an obvious, that's what a charge is. A charge means zap, right? This is more like magnets. Magnets also have an attraction, don't they? Yeah. Positive and negatives attract. That's because they're not charged, but they pull on each other because they have some, it's very much like the difference between what's happening here and what happens when you have a full charge. The opposites attract. Opposites attract. Keep that in mind. This is going to be incredibly important. If you understand this, you'll understand so much in biology. We call these types of bonds polar covalent. Covalent means sharing electrons. Polar means it's not fair. They're not sharing evenly. They're having a partial charge. That means there's a partial charge. All right. So that's how atoms get around the rule. The rule of not wanting to be charged and still wanting an outer shell that's filled. Now, what if, and I'm picking water because next week we're going to break water apart and put it back together again. All right, next week you're going to take water in a test tube, you're going to break it into hydrogen and oxygen, and then you're going to explode it back into water. You are going to take it apart and put it back together. So pay attention. Make sure you have your lab contracts and your lab safety test done by next week, Thursday. So don't miss a day. Are we clear? Yeah. All right. We're going to get beyond that. You're going to listen to what I'm saying. By next Thursday, we have to have that done. Is that clear? All right, so what if hydrogen, though, instead of making a bond with oxygen, here's this hydrogen that has one single electron, and it meets up over here with another hydrogen that is a single electron. What could it make? Pay attention, because guys, what I'm talking about now is the reason why oxygen's a gas at room temperature. It's the reason why water is a liquid at room temperature. The reason that all this happens is sitting right in front of you. This is like a very fundamental natural law, natural phenomenon, natural pattern. You really have to think about this, and then there are consequences to this. There are real serious consequences to this. So hydrogen comes together and they form a bond, right? Because they want to fill out a shell. How many electrons do each of them need? They need one each. So if they share these two here, let me use blue. If they share these two here, that means that this hydrogen has what? 1, 2 electrons. It's neutral. It has a full outer shell. This hydrogen has 1, 2 electrons. It's neutral. It has a full outer shell. They're both very satisfied. We call this hydrogen gas. We draw it like this. H, single bonded to H. Or you can write it, another way of writing it, you can write H2, gas. It's a gas. I'll explain to you why probably in 5 or 10 minutes, but we'll see how the questions go. So when they form a bond between them, they form this thing called hydrogen gas. Now, somebody asked about 1 line or 2 lines. What if you had carbon dioxide? How many carbon CO2? How many oxygens do you have? 2. How many carbons do you have? How many electrons does carbon have in its outer shell? I have to go all the way back. Carbon has how many electrons in its outer shell? 4. You go by this, it says 14, but you ignore the 1. There's a reason for it. You're just looking at the ones place. Only look at the ones place. So you have 4 here, there's 5 in the outer shell here, there's 6 in the outer shell here, there's 7 in the outer shell here, and there's 8 in the outer shell here. So this tells you how many in the outer shell. Are we clear? Carbon has 4 in the outer shell. Let's go back. Let's draw them. 4 electrons. 1, 2. These dots are the electrons. 3, 4. How many does oxygen have in the outer shell? We said it a minute ago. There's 6. 1, 2, 3, 4, 5, 6. Both of them have 6. 1, 2, 3, 4, 5, 6. What could these 3 atoms, we know they form a molecule. There it is, CO2. What could we do to these, ladies in the back? How can we take these and get a full outer shell and not have them charged? By the way, all 3 of these are neutral. When they're neutral, this is what's on the outside of their atoms in their outer shell. Well, we're going to form bonds, correct? So let's see how we can draw these. If we drew if we shared these 2, then how many electrons does this atom have? 1, 2, 3, 4, 5, 6, 7. Geez, it needs how many? 8. How many does everything except hydrogen need to fill its outer shell? 8. Okay? So let's count them again. 1, 2, 3, 4, 5, 6, 7. Needs 8. What's it going to do? Bond another one. Make another bond. Now it has how many? 1, 2, 3, 4, 5, 6, 7, 8. This one's done. It's neutral and has 8 around it. How many does carbon have? 1, 2, 3, 4, 5, 6. How many does it need? 2 more. Needs 2 more. Correct. So now let's go ahead and draw those bonds. So if it shares these, there's 2 this one's going to need to move. Let's move this one. And it can share this electron because this one's not paired. This one can move over here, right? And then it's going to form a what? A bond here, right? Anything that's unpaired, they can share. Anything that's paired, it's harder to share those. Okay? So now let's redraw this. It looks a little confusing, correct? So I'm going to redraw it. When they're done bonding, CO2 looks like this. How many lines do you see? 4. If you had oxygen, 2 oxygens, again, each one of them having 6 electrons. How many bonds is it going to make? 2. So it can share, so it can get 8. So this would look like, oxygen looks like that. This is oxygen gas. That's O2 gas. Not CO2. It looks a little messy. So that's O2 gas. Looks like that. All right. CO2 looks like this. Hydrogen gas looks like that. Single bond here, double bond here, 2 double bonds for carbon up here. Carbon has to form 4 bonds. Has to. What if you had ozone? Or no, let's not take ozone. Let's think, anyways, you get the point, right? You could have triple bonds. You could have something, you could have 2 carbons. Oh, this thing. You could have 2 carbons triple bonded to each other with a hydrogen on each end like this. As long as you what? As long as they have 8 electrons in their outer shell and they're not bonded, you can do that. You can even do 4 bonds, although that's harder. Are we all clear on that? That's what these lines mean. So when you're looking at this work, and we start looking at, let's go ahead and start looking at, we're all made of carbon. These are polar, as I said, so there's 2 kinds of things that we're looking at. Polar molecules. They have to be molecules. The easiest one to think of is hydrogen, which means this is partial negative, and this is going to be a partial positive on both of these. There's a lot of these, and if you have a bunch of these, I wish I could just multiply these by some number, but I have to actually just do this. Why is it doing that? Okay, that's my bad. So, So when we look at these and we look at water, there are consequences to all this. Oh my God, are you kidding me? There are consequences to all that. No. All right, so there are consequences to these things, and what happens is, in order for something to be a gas, it has to leave its group, it has to leave the other atoms in the substance and go off. So when you're looking at water in a pot, so let's call it a glass pot, and you got water in it, and you're heating it up. In the water, what you have is this going on. The water molecules attracting each other. By the way, this attraction, we give it a special name because it's hydrogen and oxygen, hydrogen and nitrogen. We give it a special name. It's found a lot in biology. It's the reason DNA is the way it is. It's the reason proteins have their secondary, tertiary shapes. And there's an attraction between the partial negative and the partial positive. That attraction leads to something that's unusual, some very unusual properties of water. It's a weak, by the way, where are the molecules here? Is this a molecule here when I'm circling? Is this whole thing a molecule? Is this oxygen a molecule? Where's the molecule? When I say molecule, what does that mean? The H2O, that's right. Let's be clear what the word molecule means. I think there's a lot of misconceptions about what the word molecule means. Molecule. It's a covalently bonded. Period. It has to be covalently bonded. Is water a molecule? Yes. It is covalently bonded. See the lines? That means they're covalently bonded. They happen to be polar covalent, but it doesn't matter whether it's polar or non-polar covalent. It matters only that it's bonded covalently. So water's in here, and when you heat it up, what happens to water? Once you heat it up at 100 degrees Celsius at 1 atmosphere pressure, it boils. It starts to form gas. What that means is that somehow you've added enough energy to get the water molecules. Now, when water leaves and turns into gas, it's still water. It still looks like this. It's just now in the air, rolling around, flying through the air. These things can be gases, just like CO2 and oxygen are gases. This can be a gas. Only when they are vibrating, only when they have a specific amount of kinetic energy. They have to have enough kinetic energy to break this bond. I'm just drawing two of them. These water molecules all hydrogen bond to each other. They form a matrix. So how can I describe this? How big is an ant? How big is an ant? Let's say it's even the biggest ant, probably big. Well, the biggest ant's probably pretty big, but ant is about this big, right? How big is a water buffalo? Huh? Very big, much bigger than I am even, right? They're very big. Ants can kill and carry away a water buffalo. How's that possible? They get together. There's a lot of them. It takes a lot of them. They swarm. And you'll see in Africa, you'll see when specific ant species swarm, you'll see everything in maybe South America, I can't remember, but you'll see them running away. Everything runs out of their way because these ants, when they swarm, anything that gets in the way gets taken down, broken apart, and carried away. Have you ever heard that song, an ant can't move a rubber tree plant? You ever hear that song? I'll play it to you as you leave. An ant is very small, but when you put them together, they make a very strong force. Bees, a single bee is a small sting. But a beehive can kill you. Why? Because there's a lot of them. So one hydrogen bond is not that big a deal, But when you have billions of hydrogen bonds, it becomes a very significant force. This is a molecular, is this a molecular bond, this thing called a hydrogen bond? Maybe it's a different color. Or not, because this thing wants to. Is that this hydrogen bond? What kind of bond is it? It's an attraction, but between what? It's right there, right in front of you. What is it between? Hydrogen and oxygen is correct, but is it just hydrogen and oxygen? What's this oxygen connected to? So this is a what? A molecule. This is another molecule. So it's in between molecules, right? Hydrogen bonds are bonds between molecules. Hydrogen bonds are bonds between molecules. We call them interstate, intermolecular bonds or forces, all right? These forces of attraction in between molecules determine the boiling point, the freezing point of any substance. The reason that oxygen is a gas at room temperature is that its boiling point is very, very low. Why is its boiling point very, very low? Because it doesn't have any hydrogen bonding. Water has a lot of properties that it can be attributed to the force between the molecules. So you have to add a lot of heat. A lot of heat. How hot does alcohol have to get? Does ethanol have to get? Ethanol is a bigger molecule. Let's take a look at ethanol for a second. C, double bonded C, OH. I believe, if I remember correctly, don't quote me on this, but I think it looks something like this. So ethanol is an alcohol. It has two carbons, one hydroxide. What boiling point? You use it all the time. You put it on cuts, right? Yes, you've at least seen it before. You smelled it. How? If you pour some on your hand, what sensation do you have? Well, if it's on a cut, yes. Cool. It dries quickly and it feels very cold. Always, you ever notice alcohol always feels cold? Pour it actually is very refreshing. Well, the reason why alcohol feels cold, the reason why it dries so quickly, ethanol, has a very low boiling point. It boils at 70 degrees Celsius. It turns into gas at 70 degrees Celsius. So why does it feel cold? Because when you're looking at the surface of your hand and you pour this alcohol down on your surface, the heat from your hand, remember the law of entropy, right? The heat travels from where it's warmer to where it's cooler, right? The heat from your hand goes into the alcohol. So that cools you off. And as the alcohol gets warm enough, shakes enough, vibrates enough, the alcohol goes into a gas form. That's how sweat works, right? That's how you get cool when you sweat, because water evaporates. But the cool thing about water, it can take more heat away from you. So if you did the same thing with water, this happens at 70 degrees. It takes 70 degrees. Oh, by the way, let's talk about a little bit about temperature. Temperature is a measurement of what? What do you know about temperature? Is a measurement of what? No? Oh, yes, a little bit, yes. But specifically what? It's not heat. It's interesting, isn't it? It's not. It's a measurement of average kinetic energy. It's kind of crazy. It does make a lot of sense if you start to think about it. But temperature is a measurement of average kinetic energy. So if you had water here, same amount of water, it takes much more kinetic energy to get this to evaporate, to turn into gas. So what happens is because you use water, not alcohol, you actually increase the amount of heat that leaves your hand or face or armpit or whatever. Your more heat is transferred. Because it takes longer for the water to evaporate, for it to raise its temperature, its kinetic energy enough to leave. It's 100 degrees Celsius, at least in Cleveland. It changes with pressure. I don't know if you knew that, but I don't have time to go into that's in physics and chemistry. The more pressure you have, the you change the boiling point. But here it's 100 degrees Celsius. The amount of heat that it takes to evaporate something, we call it the heat of vaporization. All right, I'm sure you covered this in AP chemistry or college chemistry, but the temperature it can change because of pressure. The amount of heat required to make it turn into gas doesn't change. That has to do with how much intermolecular forces are involved. All right. So that is that's in essence why water boils at 100 degrees, why water is liquid at room temperature, why ethanol feels cold on your skin, quickly dries, why water is, let's talk about the stickiness of water. By the way, when water sticks to itself, that process of sticking to itself, it's a general process called what? Does anybody remember? I heard somebody say it again. Adhesion is a good word. When it sticks to itself, we call it cohesion. But when it sticks to other stuff, it's adhesion. So adhesion is when it sticks to other stuff. Water does both. Water sticks to everything. Water is incredibly sticky. Everybody remember that? Everybody know that? Right? Water's very sticky. You have to know that because that, again, is one of those fundamental things that it helps you understand what's going on around you. Do you ever wonder why you get out of the shower, the water doesn't just roll right off you? You go through a lot of effort to dry, don't you? I know I do. I might use more water than you, but still. You have towels, you have hair dryers, you have there's all kinds of things you do to get yourself dry. Because water will stick to you like it's stickier. It's as sticky as tape, right? Good thing is it sticks to dirt is just as well. But adhesion is when it sticks to other things. So cohesion is when water sticks to itself. Adhesion is when water sticks to other things. That's right. It's not just water, but in this case, in biology, we're really, really focused on water. Water's for planet Earth, water is the most important molecule when we're talking about like the chemistry of. It's involved in just about everything we talk about in biology, so it's incredibly important you understand what water is. So a molecule is covalently bonded. Its intermolecular forces help define boiling point and freezing point. They can stick to each other at different rates depending if something is what we call polar or is it non-polar. And we talked a little bit about solubility and the ability to dissolve something, whether something dissolves well or not. And so when things that are ionic or things that are polar will dissolve in each other very well. Dissolves, by the way, the word dissolves, another kind of chemistry throwback, is not a chemical reaction. When something's dissolved, it is not a chemical reaction. You're not rearranging the atoms. Does that make sense? All right. When you dissolve something, what you're doing is you're mixing it. Dissolve means to mix something. So when you create a solution, what you're doing when you're creating a solution is mixing it really well. A homogeneous solution is something that looks like one thing. A heterogeneous solution is a mixture where you can see the different pieces and parts. But the bottom line is it's a solution. It's a mixing. So when I say salt is dissolved in water, I mean you put NaCl, sodium chloride, in here, and sodium and chloride are in the water. But what really happens is, why does it disappear to a point? Why is it the sodium looks like it's gone, even though it is definitely there and we could actually measure it? Well, the reason is you have sodium that has a plus charge, is a full charge. How's water going to orient itself around it? What's water going to do when you put this charged particle in it? Remember, that's right, it extracts it, exactly. These are attracted to each other out of water. In water, what happens is the oxygen surrounds the sodium because of the charges, and the hydrogens point outward, but then the hydrogens surround what? The chlorine. So when we talk about dissolve, everything's still there. Everything still is the way it was. It's just separated. These are partially positive. These are partially negative. And so they point towards the positive. The partial positives point towards the negatives. And they get separated in the solution. Once they're all filled, all the spaces are filled, if you add any more salt, where does it go? You've done this with sugar. I know your brothers and sisters have done this with sugar. You've done this with sugar. You're making Kool-Aid. Your little brother, it's usually the little brother, I don't know why. They put too much sugar in it. What happens to the extra sugar? It stays in the bottom. It just piles out. Because all the water's filled up. It's filled up all the spaces available at that temperature. We call it saturated. It's a saturated solution. So when your solution is saturated, you filled in all the possible spaces. How can you increase the amount of space in a solution to put more into it? Well, you can heat it up, and that makes the molecules vibrate, they separate. You can fit even more of the solute. We can do something called a supersaturated solution. In any case, that's all chemistry. It's not necessary for biology, so let's just move on. I just want you to understand what a solution is, because what is blood? What is blood? A solution. What is air? What is air? What is air? Come on, you can do it. What is air? I'm gonna keep saying it. What is air? A solution. Yeah, brah. That's exactly what I'm saying. Air is a solution. Blood is a solution. All the things that you consider being part of life is a stream, lake, the ocean. All these things are solutions. There are things dissolved by water in them. So it's important that you understand some of these basic concepts. I thought it was important that we review that before we move on. Is that okay? All right, so let's move on and talk about this beautiful thing. Here are the 3 molecules of life. You guys got this yesterday, I think. Yeah. Is that right? Yeah. Okay. I've made these mistakes before. Here's the difference between glucose, galactose, and fructose.
This one's the same as the Yeah, this one's the same.
Oh, really?
We got the one that says sugar 1 and sugar 2. It doesn't say.
Oh, let me see. I'm sorry. Well, let's just do this one then. Let's just take notes on this one. I'll print it for you on Monday. We got a lot to do, so I already have this one set up, so let's just do that. Sorry, messed that up. So, but it's still useful to look at this. Let's take a look at this. Let's do this together. And again, I'll give you the hard copy on Monday, and you can do it. We'll do it in class.
What is the title?
It's the same concept, but it's another different project. Biological molecules, building blocks of life. All right. So let's take a look at carbohydrates first. Here's 3 monosaccharides. Monosaccharides. Why do we call them monosaccharides? And there it is. There's only one. We'll look at polysaccharides in just a minute. So tell me, what's going on? Oh, it's just... All right, so mono means one, saccharide means sugar. Let's do a comparison. Let's take a look at it. Let's say you group from you, I'm pointing that side of the room, that 1/3 of the room, raise your hand. All high. From here, in that direction, you guys count how many carbons and how many oxygens and how many I'm sorry, let me undo that. How many hydrogens and how many oxygens? That's what I want to know for glucose. From you to you guys, I need you guys to count how many carbons, how many hydrogens, and how many oxygens. Go ahead. Start counting. Sorry, I'm sorry. And then the rest of you guys on this side, count the same thing for these. Go ahead. And write it down. I heard 6, 12, 6. If you remember the rule, 1 to 2 to 1 ratio, and you know there's 6 carbons, everything else falls into place. What about galactose? Same. C6, H12, O6. What about fructose? Same. C6, H12, O6. They're all C6, H12, O6, but they have different arrangements. Look at this. This is OH. This is the same. This is down. That's the same. This one's down, but then this one's what? Up. That makes a big difference. Not to me and you, but to your, well, it does to me and your body, but to the chemistry of these two, the chemistry of these two things is different. Just because this one points up instead of down, just because when it was forming, instead of twisting in a certain direction, to the right, it twisted to the left, that's it, done, it's different. This is almost the same but instead of connecting oxygen to the end carbon, it connected oxygen to a middle carbon, to this carbon here. This part sticks out. That changes this from glucose or galactose to fructose. This is a 5-ring structure. It's still a hexose, but it's a 5-ring structure. It forms a pentagon. It's a hexose because it has 6 carbons. But they're very different. This does damage to your liver if you eat too much of it, fructose. Glucose is not a big deal for your digestion, but can obviously lead to diabetes, and diabetic people like myself have a hard time dealing with it. Getting it out of the blood, we digest it fine. Galactose can be something that could be hard to digest if you're missing a specific enzyme. So when we're talking about molecules, their shapes make a difference. As I drew yesterday at the end of the day, I drew keys, and I reminded you that if your key just changes slightly, it works in one lock, but not another. So you have to be careful. These small changes make a big difference. So that's a carbohydrate. Those are 3 monomers, and we'll look at polymers in a minute. This is a lipid. You almost want to say it's a polymer. This is a triglyceride. Each of these is a fatty acid. A fatty acid. A fatty acid is an acidic group. Why do I call it an acid? I know my functional groups. And because of that, I know what carboxylic acids look like, what the carboxyl group is. The carboxyl group is a C double bond O. Where do you see that anywhere in here? If you're lost, it's because you don't remember what happened 20 minutes ago or half an hour ago when I wrote on the board. What's a carboxyl group? Look at your notes. You wrote it down. If you didn't write it down, you're not taking good notes. What's a carboxyl group? C, double bond oxygen, single bond oxygen. Do you see that anywhere on this page, in this lipid section? Do you see it? Yeah, I see it. Do you want me to highlight it? Has everybody had an opportunity to look at it?
Yes.
Okay. Here it is. C double bond O, single bond O. Do you see it? C double bond O, single bond O. C double bond O, single bond. Remember when you were elementary school, or when you were a little kid, you went to eat at Denny's with your mom and dad, and they said, "Where's Waldo?" And they said, the word, you had that little game, that word circle thing. You used to circle the words with crayons. You ever do that? Well, this is what you're doing here. Where's your C double bond O? Oh, where's Waldo? Well, there it is. This is called a carboxyl group. They form carboxylic acids. So this is what makes this an acid, a fatty acid. Why is it fatty? Because of this, what is this? A long chain of what? Mostly what? Carbon and hydrogen. Look at that chart I gave you yesterday. Mostly carbon and hydrogen. Also known as a hydrocarbon. Long chain, we call this fats. That's what a fat is. So this is a fatty acid because the acid. There's 3 fatty acids, so we call them tri. There's 3 for tri. When you go to a doctor's office, one of the first things they look at is your triglyceride level. You'll see it on tests. Ask your parents to look at their MyChart. You look at their blood tests, and one of the big blood tests they do is a triglyceride level. This is a triglyceride. Now, why glyceride? Why is this a thing? Well, it turns out that this little tiny, let me use white, I guess. Here, this 3 carbon, usually OH, You know, there's probably an OH here. Probably drawing this wrong, but I'm just gonna go ahead. This thing here is called glyceraldehyde. You'll see it pop up throughout the book reading. You'll see it pop up in all of biochemistry. These three little carbon, in fact, this is what plants make. They put two threes together to make a six. What have you heard of that's six? 6 carbons, 2 threes equals 6. What could a plant put together? 2 threes to make what? 2 threes can make a 6. So plants will take these will make glyceraldehyde, and they'll end up making a glucose molecule. All right, so it's called glycerol. Oh, there it is. They actually had it here already. I shouldn't have called it glyceraldehyde. It's a glycerol because of the alcohol. Sorry about that. So there it is. It's actually already drawn for you. Glycerol. It's drawn for you here. 3 carbons, 3 OHs. Fatty acids are any of these long chains. You can see we don't have to, obviously nobody wants to draw these out. So what you do is you put them in parentheses, CH2 times how many? 12 of them, with the end being a CH3. That's 1 fatty acid. That's probably this one because it's the smallest one. Here's CH2 times 7 with more CH groups, another 7, so there's a branch here with the end cap being a C. So if we drew it, that's this one here. What is the difference between this one here and this one here? This one and these two. What's the difference? That's right, the double bond versus the single bond. Do you see that? Where's Waldo? I found him. So double bond versus a single bond. If you have a double bond, we call them unsaturated fatty acids. If they are all single bonds, we call them saturated fatty acids. When the fatty acid is unsaturated, what that means is, let's look at what they look like when they're saturated. That means that all the bonds have a hydrogen. That's what saturated means. They're full. All the carbons have a hydrogen on them. All the bonds have a hydrogen or a carbon or an oxygen. There's no bonds. They're just kind of forming 2 bonds. If you have an unsaturated, what happens is you have C, you have a double bond somewhere, so that you have a carbon You have a carbon that does not have a hydrogen attached. So they're unsaturated. Well, what's the big deal? Well, there's a big consequence to that. These saturated fatty acids, they form long chains that can layer one on top of the other. Now, they don't form hydrogen bonds. They don't. But because they're heavy and they're long, they're very long. As you can see, they're 20, 30 atoms long. They don't easily turn into gases. It's not like carbon dioxide or oxygen that are small and nonpolar. These are very big and nonpolar, so they don't turn into gas easily. And because of that, they can lay down, water can be surrounding them or not, they can lay down in flat sheets and they form solids. Where the other ones, the unsaturated fatty acids, they can start to go along, but then this makes them kink, makes them bend. And because of that, they find it harder, to lay flat on each other. They'll lay down, but they won't lay flat. So they end up, these guys here end up being mostly, they tend, I'm going to use the word tend, to be liquid at room temperature. And these down here tend to be solids at room temperature. Let's just say temp. So these tend to be butter or lard, right? Those are things that tend to be solid at room temperature because they have a lot of saturated fatty acids. Where these up here might be corn oil or olive oil because they tend to be liquid at room temperature. That's why olive oil and butter have two different states of matter at the same temperature. That's why when you're done cooking with corn oil or olive oil, the pan cools down. What's in the pan? When you're done frying something in corn oil, what's in the pan after it cools down? It's a liquid. What's in a pan if you cook with butter after it cools down? Or you fry a pork chop in a pan and you take your pork chop, you go eat it, the pan's time to wash the dishes. What do you find in the pan? What is it? Solid. So the difference is one's saturated, the other's unsaturated. Now, health-wise, how does that make a difference? It turns out, I'm really simplifying this, but it turns out that these can lay on each other very easily in your blood too, and they can start to lay and form these things called plaques. They get chemically changed. So if your blood vessel looks like this in that bloodstream, over time, as you're eating saturated and other types of fatty acids, the theory is that these things turn into these little plaques. They start to do what? Start to block your arteries and veins. And they can block tremendous amounts before they become a problem. And then you get a small blood clot because somebody punched you in the nose, or you bumped your knee or something, and that blood clot comes along and it gets stuck in that artery or vein. So that means you don't get any blood flow through that vein or artery. We call that a thrombosis, right? It could be a heart attack. It could be a stroke. It could be a lot of things. So as you get older, if you've been eating a lot of these, it has to be a lot. It turns out saturated fatty acids is good in small amounts. We need it, but it's the unsaturated fatty acids that actually tend up to be the good kind of fatty acids you need to be eating. So you reduce the tendency of your body to form these plaques. So I'm hoping I'm making a connection for you why this is important. This idea of saturated versus unsaturated fatty acids. Now, proteins are made of amino groups, and this is a drawing of an amino group. The amino group is here. I'm going to identify, here's Waldo. There's the amino group. There's an amino group. There's an amino group. And it says amine group. I'm sorry, amine group. What's on the other side? What's on the other side? Carboxylic acid group or carboxyl group. So I got amine on one side. I got an acid on the other side. What am I going to call this? Amine on one side, an acid on the other side, amino acid. You need to know what these look like. Sticking off the side is a variable. It's not an element. It's a variable. We call it the R group. It sticks off the side. There's 20 different possibilities. There's 20 different R groups here. Okay? If it has a carbon with a sulfur on it, we call it cysteine. That's the amino acid cysteine. If it's just a carbon and 3 hydrogens, here for the R group, we call it alanine. This is nonpolar. This is small. This can form sulfur. If you have a lot of cysteine in this protein that you call hair, then your hair is kinky. The more cysteine you have, the more genes you have that say put more cysteine in this protein, right? It's a gene. Then the curlier your hair, the kinkier your hair. If you have less cysteine in that protein, then your hair is straighter. That's it. That's the only difference between really curly hair and straight hair is how much cysteine you have in it. Because the sulfur makes these types of bonds that are like hydrogen bonds. We call them disulfide bridges, but in any case, that's the point there. So which amino acid? There's 20 different amino acids. There's amino acids that have ring structures. There are amino acids that are acids. There are amino acids that are bases, basic. There's amino acids that have formed these aromatic rings. They're really large amino acids, really small the smallest amino acid is the same structure, the amine group, the carboxyl group, the H group. And here you have an H, it's alanine. It's the smallest amino acid. So there's 20 different ones. That allows proteins to be very complicated. It would help you understand what's going on if you were paying attention to this instead of being on your phone. That would be very useful. Now just focus on the job in front of you. All right. Here is the sugar ribose or deoxyribose. Which is this? Ribose or there's 2 possibilities. It could either be ribose or deoxyribose. It's not easy to tell. You probably won't remember. I don't think I mentioned it recently. You're looking at this carbon right here. You're looking for what? What are you looking for? Whether or oxygen or no oxygen. If it's no oxygen, it's what? Deoxy. If there is an oxygen, then it's ribose. All the oxygens are usually written. The hydrogens, most of them are never written in these drawings. Okay? Which of these is it? Deoxy. This is deoxyribose. This is DNA. All right? So we're looking at DNA. DNA nucleotide is made. These are monomers, by the way. These are monomers. You can put these together to make disaccharides or polysaccharides. These are the monomers for proteins. You can make long polymers called proteins or short polymers called proteins. There are dipeptides or tripeptides and polypeptides. And nucleic acids obviously form you put a bunch of these together, you get billions long, called DNA, right? A DNA nucleotide. This is called the nucleotide. And there it is, it says nucleotide, right?
How would you describe the position of the oxygen in a ribose?
Good question. So with all ring structures in chemistry, you start with the 0. With at least organic chemistry. You start with the 0, all right? And you go around clockwise. This is position 1 prime. This is position 2 prime. This is position 3 prime. This is position 4 prime. And this is position 5 prime.
So you would say on the 2 prime?
2 prime is correct. And that's how you number these. There's only 5 carbons, by the way. Both ribose and and deoxyribose are what we call pentoses. There are only 5 carbons. All right? On the 5', this is really important because it turns out that because there's 2 different things happening, because you're gonna, and I'm gonna draw it out here as a block in a minute. You have, over here you have the phosphate group, that's part 1 of a nucleotide. You have the ribose, the sugar part, that's part 2 of a nucleotide. And then you have the nitrogenous base, that's part 3. Do you see why it's called a nitrogenous base? They call it nitrogen base. Why do they think they call it a nitrogenous base? It's really simple. The questions I'm asking are generally simple. Why do they call it nitrogenous base or nitrogen base? Because it has nitrogen in it, and it's a base. That's all. Honestly, scientists, although we try to be imaginative, or generally try to be really simple. Yeah?
Would you say that, then would you say that it's part 2 or the second?
Someone close that door, please. What is this?
Describing the position of the ribosome. Are they all, is that,
This would be, what I would say is the nitrogenous base is on the first 1' of the ribose. Always. The base is always attached to the 1'.
I thought you said it was the sugar, the middle one was the sugar.
This is the sugar, yeah.
So it's always on the sugar?
It's always connected to sugar, yeah.
Okay.
The base is always connected to the 1' of the sugar. On the 2', I can look for the oxygen to determine if it's a ribose or deoxyribose, but it's really not involved in any kind of chemical reaction. It's just having the OH or the H forces, because of geometry, forces the nitrogenous base to be either T or U. That's the only issue with this 2nd carbon. Then this OH is always on the 3rd. This is incredibly key. And the phosphate's always on the 5'. So when we get into DNA replication and DNA structure, this becomes very important. Let me show you why. So you have this phosphate group. It's attached to a carbon, right? That's attached to, as someone just asked, to a ribose or deoxyribose, in this case deoxy because it's sugar, which is then attached to a hydroxide group, which can then be used to attach to what? Can anybody make a guess? What would you attach then to? What would you attach to this? Actually, this leaves. What would you attach to this? I'm just going to go ahead because we're running out of time. Another phosphate, which is attached to what? Another carbon, another ribose or deoxyribose, which is attached to what? A phosphate, which is attached to what? Another deoxyribose, which is attached to a phosphate, it's attached to another deoxyribose, and then get attached to another phosphate. Yeah, I was gonna keep saying it till a lot of people started complaining. That's all. Yeah, it's a chain. It forms a really long chain. I mean, really long. When you leave the room, you can look outside and that beautiful poster that I bought so long ago, and it's just this long chain. It's just this long chain. All right, but because there are 2 sides, because there's a 5' and a 3', because they always attach this way, because you're always attaching the phosphate to the 3' of the previous nucleotide, because that's the case, you have a 5' side to the molecule, all right? And you have a what's on the other end? What's on this end? A 3 prime. These molecules have direction. They go from 5 prime to 3 prime. That's how you attach them. What does that allow us to do? My God, guys, this is amazing. This is so stunningly crazy that because there's direction, you have order, you can write a book. This becomes a language. Because there's differences in between them in the nitrogenous base, you can have an A, the nitrogenous base B, T, the nitrogenous base G, the nitrogenous base C. It can be anything depending on, and it can be billions of nucleotides long. Now you have a book, which then can be defined into genes, and then genes can make someone tall or short or have wings or have be a jellyfish or a fish or an octopus or a human or whatever, because there's a series of letters that has direction. If I had a bunch of letters and there were no direction, I didn't know which was left, right, which way to read it, then there's no way to have a language. You can't have a language without direction. You can use anything to form a language. We're used to using letters, but in Arabic, the letters are more like script, right? Like drawings. In Chinese, they're symbols that mean ideas. They don't even mean specific words. You put the different symbols together to form different words. Like, I don't know. I don't know Chinese, but I know that much about it. So the same in Japanese. In Korean, they're more like letters. In the ancient Mayan calendar, when the conquistadores, my ancestors first came, some of my ancestors first came to the New World, and they killed the Native Americans and took over the Aztec temples and we killed everybody. We killed them with this. We gave them smallpox blankets. We shot them. We wanted the gold. That was it. Sorry, you're dead. We want it. I'm not saying it was a good thing. I'm just saying it did happen. So bottom line, when some of my ancestors came here, they brought their priests, and the priests tried to figure out what the heck was going on in the temples, and they went to the libraries. They found a bunch of strings with knots in them hanging on sticks. What do you think that was? It was their language. They were their books. Their books were written in strings, top, down, outer, in. Direction, order, gives you words or math. They kept all their math there. An abacus is not numbers. They don't use numbers. They use little beads on a string, and they move them around. And they, in their minds, it means the same thing as 1003 for you. So language can come in chemistry as well.
Can you zoom in on the paper at the bottom?
Oh, I left this like that. Zoom in where? The paper at the bottom.
Yeah.
All right, good. I know it was a lot, but I think we had a good class.
Can you do a little bit more?
Huh?
A little bit more?
Yeah. I know it was a lot. Thank you for your patience. I'll try to minimize these days, but it is AP Biology, guys. There's a lot to cover. And it's either you read it on your own or I explain it, and me explaining it means language. Sorry. So here it says, use Model 1 to show which atoms are present in each type of molecule by listing the symbol for each atom included. Carbohydrate has been done for you, CHO. So if we think about it, carbohydrates are CHO, but remember let me go ahead and just kind of put it off to the side here. The carbohydrates are one carbon to 2 hydrogens to 1 oxygen. I don't like that. It looks like a 10, but so let me make it one oxygen, right? So one carbon to one hydrogen to one oxygen. So that means, again, for every one carbon, you're going to have two hydrogens and one oxygen. All right? So remember that. 1 to 2 to 1 ratio. In lipids, you do have some, you're mostly, and let's just go ahead and say mostly carbon and hydrogen. You do have, and I'm going to put in parentheses here, you do have sometimes you have phosphorus. First of all, you do have some oxygen. There's some oxygen. Sometimes there's some phosphorus and even some nitrogen sometimes. But these, the nitrogen is very rare compared to the others. There's obviously a lot of oxygen, and phosphorus is in certain kinds of lipids. But for the most part, when you're talking about lipids, you're talking about carbon and hydrogen, long chains of carbon and hydrogens, or a ring of carbon and hydrogens, mostly carbon and hydrogen lipids. Amino acids, you're dealing with carbon and hydrogen and nitrogen and some of the R groups, and of course you have oxygen and and the amino acids, some of the R groups have sulfur in them, but no phosphorus. This is one of those key points. Though lipids do have phosphorus, not all lipids do. There is no phosphorus in amino acids. And there's plenty of nitrogen in amino acids. Plenty of nitrogen. Nucleic acids also have a lot of nitrogen. So you have, with nucleic acids, you have carbon, you have hydrogen, you have oxygen. That should be fairly reasonably understood by now when you think about the fact that nucleic acids do have that sugar, either ribose or deoxyribose. They have the nitrogenous base, and then they have that phosphate. All right. So remember, that's the nucleotide, that's the monomer sketched out there. So when you're dealing with nucleic acids, you're going to have carbon and hydrogen and oxygen because you have a carbohydrate as part of the structure of the monomer, but you also have nitrogen and phosphorus. And let me rearrange that and change that color and say nitrogen and phosphorus. So when you see nitrogen, you're going to find nitrogen in places like amino acids, nucleic acids, and even lipids sometimes. But you are definitely going to find phosphorus mainly in nucleic acids. Obviously, lipids, phospholipids have phosphorus in them, but you're really going to find a lot of phosphorus in nucleic acids because it's part of the structure of nucleic acids of DNA and RNA. Amino acids are mainly the main identifier is nitrogen, but then it's going to be hard to tell the difference if you're just looking for nitrogen between amino acids and nucleic acids. It's the sulfur that's going to be the key. If you find sulfur somewhere, then you're probably looking at amino acids or proteins. Now, I'm not going to go back and go over all the components. You have all the notes for all the different monomers, but you should know for the exam, like
for the biomolecules, of lipids, nucleic acids, and proteins and carbohydrates, you should know the monomer and polymer of each. So in lipids, you do have fatty acids that can form. Fatty acids do form parts of a triglyceride, but honestly, you have to say there are no monomers or polymers, because when you're talking about a polymer and a monomer, you're talking about taking like a Lego, like I keep using the Lego blocks as an example, taking a bunch of Lego blocks and putting them in locking them together. In the case of molecules, it would be covalent bonds. But taking these Lego blocks and linking them together to form something bigger, lipids don't do that. They're not you can't make these giant long molecules like that. Lipids are just carbon, hydrogen. They don't really have monomers or polymers. Nucleic acids, I hope by now you know it's a nucleotide. At this point, the test is on Monday. I'd expect you guys know that a nucleotide is a monomer, and the polymers are either DNA or RNA. And the proteins, you have amino acids, are the monomer, and the polymer are polypeptides. And there's whole long days of lectures and information. I told you to make index cards on all of these. This is stuff that you need to know. Carbohydrates, there you talk about monosaccharides. Let me zoom in and write it. So you talk about monosaccharides, and the polymers polysaccharides. The monosaccharides are, remember, glucose, galactose, fructose. Those are the ones we talked about in class. And of course, ribose, and of course, deoxyribose. Remembering that deoxyribose and ribose are, let me go ahead and separate them. Deoxyribose and ribose are pentoses. There are 5 carbon sugars, and fructoses, galactoses, and glucoses are 6 carbon sugars. They're monomers. Now, polysaccharides, you also have some disaccharides that we've discussed. And some of the disaccharides are table sugar or something called sucrose. Do you need to know that sucrose is glucose and fructose? If you know that, that's great. You do not have to know it for the tests. I'm not going to ask you such a question. And, lactose is another disaccharide we talked about. Do you need to know that it's glucose and galactose connected together? No, not really, but it's nice that you know it. It'll help you in the long run, but in the years to come. But right now, for this test, no, you do not have to know that. You should know that sucrose and lactose are disaccharides. It means two sugars linked together. Two Lego blocks. The prefix di means two. All right, so two. All right, whether it's glucose and fructose, or whether it's glucose and fructose, or glucose and galactose. If it's glucose and fructose, it makes sucrose, table sugar. If it's glucose and galactose, it's lactose, it's milk sugar, etc. And then the polysaccharides we discussed are cellulose, which is, you call it wood, right? Paper, pencils, wooden structures. The sugar made for structure in plants, not digestible by anything except for bacteria and fungi with special enzymes. It's the only way to break down cellulose. It's so carefully put together. Another polysaccharide is starch, and that's found in plants. We eat it. And finally, another polysaccharide is going to be glycogen, and that's in animals, in the liver of humans and mammals, but in animals. All right, so 3 polysaccharides we discussed: cellulose, starch, and glycogen. And then you have 2 disaccharides we discussed as sucrose and lactose. You should know that cellulose is in plants, that it's for structure. You should know starch is in plants, it's for energy storage, and glycogen is in animals, or energy storage. That kind of thing we should know, and that's kind of a quick, and yet one more quick review. But if you wanted to tell the difference between these 4, these are the elements that are found in each one. Which type of molecule includes the example of long-chain carbon backbone? Long-chain carbon backbone. Now it says carbon. If it said carbon and phosphate, then it would be DNA. But it says long chains of carbon as the structure upon which everything is built, the backbone. You'd have to say lipids, right? Why? Because it's carbon. It's mainly carbon and hydrogen. It's mainly carbon and hydrogen. Yes, there are other elements involved, oxygen, phosphorus, and nitrogen sometimes, but carbon and hydrogen being the main component. All right. In a molecule referred to in the previous question, which is the dominant element attached to the carbon backbone? And that it would have to be hydrogen, right? As I said, it says dominant element, so it's carbon and hydrogen, right? The carbon backbone with hydrogen attached. Fatty acid chains and lipids is often referred to as a hydrocarbon chain. Hydrocarbon, hydrogen and carbon. Discuss with your group why the chain is given the name. Write 1-sentence definition of hydrocarbon. Long chain of carbon and hydrogen. Now, I have already given you all this information in class, but again, I just want to make sure that it's all written down in order and there's no confusion about what the right answer is. Which molecule has a central carbon with 4 different components around them? Central carbon with 4 different components. Well, I drew it in class. Again, giving you all the answers. You have a central carbon. Then you have attached to that central carbon an R group. On the other side, there's a hydrogen. Then on one side, you have a carboxylic acid group. And on the other, let me use a different color. On the other, you have an amine group. So you have central carbon with 1, 2, 3, 4 different groups surrounding it. We call that an amino acid. Amino acid. Right? The amine group and the carboxylic acid group helping define that word. Which molecule has sugar and nitrogenous-based phosphate group? Now here again, I'm going to draw it out over here because there's more room. Well, I already drew it. I'm not going to draw it out again. Well, I guess I might as well. Has a sugar. Yes, there's hydrogens and carbons. It's a 5-carbon sugar again. 1, 2, 3, 4 prime, 5 prime. To that 5th prime carbon, there is attached a phosphate group. And on the other side, a nitrogenous base. Right? So as a nitrogenous base and a phosphate group attached to a sugar, what the sugar can be ribose in DNA and can be deoxyribose in RNA. In DNA. So DNA is deoxyribose, and I don't even know what I said a minute ago, but RNA is ribose. Okay? So the answer is nucleic acids. The two main nucleic acids that we talked about, the only two we talked about are ribose and deoxyribose nucleic acids. Discuss with your group members some similarities among all 4 types of molecules. List as many as you can. Well, they all have carbon, hydrogen, and oxygen, with carbon being the center component of just about every one of them. You make them all the same way. Let me be neater because I know I'm going to hear it if I'm not neat. Make them all with dehydration synthesis. That's 2 similarities. They all use the same group of elements to be formed. All of them use the same group of elements, which is schnapps, with different percentages for each one, right? Carbon, nitrogen, oxygen, phosphorus, and carbon, nitrogen, oxygen, phosphorus, and sulfur. All right. What is the chemical formula of the first carbohydrate molecule shown? Well, if you look at the first one, it's C6H12O6. 3 structural groups shown. Do all amino acids have what 3 structural groups shown do all amino acids have in common? Well, they all have the carboxyl or carboxylic acid, the amine, right? And hydrogen. They all have all 3 of those. / central carbon. The difference is, of course, the R groups. There are 20 naturally occurring amino acids. and each one only varies in the structure of the R chain. There's that R group or R chain, right? Two amino acids shown in model 1. What are the R side chains in each? Well, one is CH3. That's the R chain. That means that's, to draw it out, it would be one carbon. Let me draw it neater. Trying to be neat and move it quickly at the same time is very difficult. CH3. So you have a carbon with 3 hydrogens attached to it, sharing. Remember, carbon always makes 4 bonds. And this last one goes to the center carbon, right? So that's CH3. That's how we would note it. The other one is CH2SH. So that's one carbon. Now remember, these are R groups. So to be clear, when you look at an R group, you're looking at this group sticking off. So CH3 would be sticking off here. So you have amine, center carbon, hydrogen, carboxyl group, and then you have CH3 sticking out here. All right. So that being said, what does that look like? Well, that's going to look like, well, you have a carbon. You have to that sulfur. Here you have a hydrogen sticking out, and the sulfur has a hydrogen sticking out. So that's all. So you see the only difference between these two is that you have a sulfur here instead of a hydrogen, 3 hydrogens. Carbon still has 4 bonds. During chemical reactions, the bonds of molecules continue to be broken and reformed. To break a bond, energy must be absorbed. When bonds are formed, energy is released. If more energy is released than absorbed during a chemical change, the process can be used as a source of energy. The general rule for processes such as respiration, carbon is the more carbon atoms that are in a molecule, the more energy the molecule can provide to the organism when it is used as food. So we did an experiment in the lab. We did a lab where we took water and we broke it. So we took oxygen and 2 hydrogens, and we snapped these. We snapped these bonds, and we used electrical energy to do that. In the form of a battery, right? We inputted electrical energy, broke the bonds. Then we just added just a tiny bit of energy, And what we did was we formed water. We put it back together. When we form water, we put energy in to break water up. When we formed water, energy came out. And it didn't sound like a lot because it was just a few million molecules. When you use more material, you get more energy. But I think that's obvious. Like the difference between a firecracker and a stick of dynamite is how much explosive material you have in it, right? The more you have, the more intense the explosion. So our explosion was tiny because your parents want you back in one piece, and I want you going home safe. So we made those tiny little explosions, and that released a lot of energy. When water was put back together, I don't know why I drew it that way. Let me erase that. So when we put water back together again, right? We took that. We used electrical energy, and what we formed was hydrogen gas, and then oxygen gas. We collected it in a test tube, right? And we know that there's a 2-to-1 ratio. So this is 2 hydrogen gases for every one. So we had a lot more in the hydrogen tube than we did in the oxygen tube. And so when we made that hydrogen gas, and then we added some oxygen from the air, oxygen gas in the air, and let me just put in parentheses down here, from the air, right? And we allowed the hydrogen and that oxygen to mix, and we added just the tiniest of little sparks, a little bit of heat from our flame. We got an explosion, and then what formed was water. And it was liquid at room temperature. So what we did, what is it that one of the things you should have gotten from that, or hopefully you got from that, is that we needed here at this stage, when we were going in one direction, we had input energy. And here, when we reversed it, we got energy out. Now, if we got more energy out than we put in, then we're making energy. We can use that energy to run reactions like our thoughts, our heart beating, etc. And that goes for any chemical reaction. You're either putting energy, you get a net energy in, or you're putting net energy out. So when you have a long chain of carbon, like in fatty acids, for instance, in fats, and if they're the longer the chain, the more bonds you have to be able to break apart, to use. The more energy you have, the more of these bonds that you can break and then reform in the form of carbon dioxide. And when you do that, you get energy out, and boom, you can use that energy to run the different things that you need to do in your body. So fats are one way we can do it, but we can use water as a simple way of looking in a very simple ratio, and I think fairly dramatic way. We can see energy coming out, energy putting in, electrical energy going in, and we get out that explosive energy when we make water. Here it says, using the information above, is this a carbohydrate or lipid? More likely a good source of energy for an organism. It's going to be the lipid, right? Because the lipid has a lot more bonds that you can break. And so lipids are much better at holding energy. There's a lot more energy storage with lipids than you can with carbohydrates. So here's a second model. Here again, glucose and fructose. Here's amino acid 1, amino acid 2. In both cases, we're putting them together. All right, I've drawn this out for you numerous times, but let's go ahead and see exactly what's going on in a professional drawing here. You have OH and OH, and they're coming together and they're forming a bridge. This oxygen is going to be used to bond to this carbon, remembering that at each stage, at each one of these places where there is no C, there is a carbon. So this is carbon 1. It's actually, it's complicated, but this is actually carbon 1 prime, 2 prime, 3 prime, 4 prime, 5 prime, and 6 prime. And this is carbon 1 prime, 2 prime, 3 prime, 4 prime, 5 prime, and 6 prime, right? Now, why were they numbered that way? They were numbered that way before they closed in a ring, and after they closed in a ring, when the oxygen forms that ring with the second carbon here, it forms fructose. If it forms the ring with the first carbon here, then it forms glucose. Irrelevant, that's not the point. The point is we're going to use this carbon, we're going to connect this ring to this ring, all the carbons, I'm going to put a purple dot at each carbon that's not labeled with a C, remembering that every time there's a line that comes together, they form that is a carbon, And if you count them, each carbon has 4 bonds. 1, 2, 3, 4, 1, 2, 3, 4. You can go around and count them. Even this, it says CH2OH. If you broke it up, if you opened it up, this is kind of a condensed look at it. If you looked at this, right, at this, use a different color. And let me use green. So if you looked at this, it would look like carbon, then an OH group, then 2 H's. And then this whole thing would be attached to this carbon. So that's what CH2OH means there. So what you see is if you count them, there's a single bond, there's 4 bonds to this carbon, there's 1, 2, 3, 4 bonds to this carbon, etc. Okay then. When we look at the combination, when these two coming together, what you get is an OH and an H. An OH and an H coming together because something has to leave. Remembering oxygen can only make 2 bonds. Let me before I do that, there's a bond between the oxygen and hydrogen, a bond between the oxygen and hydrogen, and then there's a bond between this oxygen and this carbon, this oxygen, this carbon. That forms 2 bonds for oxygen, 4 bonds for carbon, 1 bond for hydrogen, right? So when we draw the line, something has to leave in order to get these 2 molecules, these 2 monomers, these are called monomers or monosaccharides, to come together. We have to have something leave, and what generally leaves in these reactions is water. So water comes out, and what gets put together is that oxygen still needs to make 2 bonds, but instead of this oxygen being bonded to this hydrogen, this oxygen now binds to this carbon. And so oxygen has 2 bonds, and each carbon has 4 bonds, 1, 2, 3, 4. And now these 2 are connected. And we name this sucrose, right? Table sugar. And of course, we got water out. Where'd the water come from? The hydrogen and the hydroxide coming together making water. Exactly what we did in the lab. All right, so when we're looking at an amino acid, these 2 are coming together. Again, if you're going to put this amino acid this, and nitrogen can make 3 bonds, 1, 2, 3, that's full. Carbon can make 4 bonds, 1, 2, 3, 4, that's full. How do you get these 2 connected? Well, you got to get rid of something, like we've said numerous times. This hydrogen, these hydroxides get together. They form water, right? They leave in the form of water. So when they're, and as they leave, in the form of water, what you see connected between the 2 is the NH group of 1 gets connected to the carboxyl group of the other, and you get carboxylic acid on one side and amine on the other, and there's a connection between the 2. We call that a dipeptide. This is sucrose. Both of them are 2 monomers connected together, remembering that a monomer of a protein is amino acids, so 2 amino acids coming together would form not a polypeptide, poly means a lot, but a dipeptide meaning 2. Sucrose is a again, the 2 again, disaccharide. Di meaning 2, saccharide meaning sugar. Now, when you're talking about lipids, again, carbon-hydrogen backbone, mostly carbon hydrogens. I think if you look at this, I think and no one could argue this, mostly carbon and hydrogen. Even the glycerol molecule is mostly carbon and hydrogen. Has more oxygen than a fat does. There's a double bond oxygen here. This carbon is a carbon double bond oxygen, single bond oxygen. So it used to be a carboxylic acid group. The H left, right, and then the OH from here left, and the H from this hydroxide left. Together they form this bond. I've drawn it all out in your notes. I'm not going to do it again. All this I've done in your notes before, but I'm not going to. I can't just redo everything, so I'm going to go on. So if you have this long chain of carbon, that's one. Now you got two other spaces. You can take this fatty acid. I'm going to go ahead and draw this out, even though I've done it in class. I just want to point out that this means double bond oxygen, single bond OH, right? Single bond carbon with two hydrogens repeated 14 times. So it would be over and over again 14 times. That's all that means. It's just a shorthand to let you know that this is repeated 14 times. So instead of doing that, I'm just going to put dot, dot, dot. Then you have the final carbon with 3 hydrogens because every carbon needs 4 bonds. So that's your fatty acid. And you're going to take that and you're going to connect it to this. How are you going to do that? How does that happen? It happens the same way as everything else. OH and H leave. Let me zoom in on that. OH and H leave. And what you make is water, right? So you put them together and you make water, so you get this, and that's a diglyceride. This is a monoglyceride, meaning one glyceride molecule. It's a diglyceride, and then you can have a triglyceride, but you're never going to have a polyglyceride, for instance, so there's no polymer. That's why I don't even want to talk about a monomer, because the max you're going to make is these 3 together. You're not going to make many like you can with DNA or amino acids or sugars or carbohydrates. So, okay, so we're talking about dehydration synthesis. What are the reactants in A? The reactants in A are monosaccharides. Or carbohydrates, if you'd like. What are the products in A? They're disaccharides. And water. Each of the reactants in A is a single sugar molecule, also called a monosaccharide. What is the prefix? Well, maybe they wanted specifics, so you could say glucose and fructose, and then here you could say sucrose, which are disaccharides, and glucose and fructose, which are monosaccharides, right? Each of the reactants in A is a single molecule, also known as monosaccharides. What prefix before saccharide would you use to describe sucrose? Well, we already said it, and they told you already. It's di, which means 2. What are the reactants in reaction B? They're amino acids. And what 2 molecules in reaction B are joined together? When 2 molecules in reaction B are joined together, amino acids are joined together, what other 2 molecules are produced? There would be a dipeptide and water. I just want to remind you that when we're talking about this, you're talking about this word peptide, what it means. It means two amino acids bonded together. This here, this bond between this carbon and this hydrogen is known as a peptide bond. And because it's a peptide bond and you have two amino acids, we call them dipeptides. That's it. So it's a dipeptide that you put together in reaction B. You can go look at the paper on that. And what product do all three reactions in Model 2 have in common? Water, H2O, as a product. Water is always produced in dehydration synthesis. When two sugars are joined together, a new bond that forms is a glycosidic bond. There's two bonds between sugars are glycosidic bonds. The amino acids join together to form a peptide bond. When fatty acids are joined to a glycerol, that bond is called, holds them together, it's called an ester bond. So they actually have names for each of the bonds. As I mentioned, peptide bond. On the diagram Model 2, circle and label the glycosidic peptide and ester bonds. So it says on the diagram Model 2, circle and label the glycosidic peptide and ester bonds. The glycosidic bond, I'm going to circle it like they ask, and I'll do it in blue. This is the glycosidic bond because it's between two sugars, between the two amino acids. I already did it up in the drawing previously, but I'll go ahead and circle it here. It's the peptide bond. And then the ester bond is going to be here between the fatty acid and the glycerol molecule here. Those are the ester bonds. These reactions can also be reversed, breaking a large molecule into its individual molecules with substance would need to be added in order to reverse it. Well, I'm pretty sure I've told you numerous times, but I'll say it again. Water goes in, breaks them apart. Lysis means to split or separate. What prefix would you add to lysis? If you're trying to separate all these biomolecules, what would be the prefix you would use? If lysis means break, what would you put in ahead of it if you need water to break them? Give me a minute. So word is hydrolysis, using water to break it. Using your answers from the previous two questions, what word is used to describe the reaction that uses water to break up a large molecule? Well, hydrolysis. Oh, I guess I skipped to the point, didn't I? Apologize. Hydro is there, that's the prefix, and together you would make hydrolysis. All right. To break water, to break up large molecules using water. So to be clear, you have a large molecule, you have some kind of reaction, it's reversible, right? It goes in both directions. You can use, you know what, let me draw two arrows here, just because I think it would work better if I did that. So let me draw two arrows. You're going to use some big molecule, and you're going to break it up into its units. Right? You're going to break up something into a biomolecule into parts. It's reversible, so you can go in this direction and break it apart. Or you can move in this direction and put them together. So when we break it apart, take a big thing and break it apart into little things, we're going to call it hydrolysis. hydrolysis, because we're adding water, goes in, MSA is used, all right? And then to build it, it's another word for building is synthesis. And we're going to make water. Water's coming out, so we're going to call it dehydration. Ugh. I hate that it takes this long. Sorry, I have to start all. I've written it 3 times. Dehydration. Dehydration synthesis uses water. I'm sorry, produces water. It kicks water out. That's why it's called dehydration. So to build things up, use dehydration synthesis. To break things down, you'd use hydrolysis or hydrolysis. You're using water to break things up. You're making water when you build them up. Build them to make something, a large biomolecule. This is a biomolecule. So you're building a big biomolecule, and these are the monomers. Now, let's not even call them that. Smaller units. Why don't I just call them monomers? Because lipids don't really have monomers and polymers. These ester bonds are broken. You do have fatty acids, so I'm not going to get into the discussion about the difference between monomers and polymers. Just know that this isn't, lipids are not true polymers, so let's not even call them that. They are molecules, but not true polymers. All right, let's move on. So as metabolism is the collective term used to describe all the chemical reactions taking place inside living organisms. So why is water so important in metabolic? I don't know what's wrong with me. Metabolic reactions. What is water? Why is water so important in metabolic reactions? I'm sorry, it's been early in the morning and I haven't had a lot of sleep, so I apologize. So, metabolic reactions, why is water important? Because it's produced when building reactions or molecules. And it's used in the breakdown of biomolecules. We store excess food in our body, either in the form of carbohydrates, in muscles, and liver, and fat, and adipose tissue, right? So carbohydrates are stored in the liver and muscles in the form of, and you should know because I summarized it at the beginning of this, but I'm going to go ahead and say glycogen, right? That's how it's stored. And in adipose tissue, it's stored as fat around your belly and different places in your body. You store excess food, excess energy in the form of fat. When your body needs additional energy, it uses carbohydrate source first. Is it quick? So it uses glycogen, which, by the way, is how our feedback loop that we talked about with glucagon and insulin works. It releases glycogen from the liver. All right. Why do you think carbohydrates are used as a source of quick energy rather than fat? Use complete sentences and your scientific terminology in your response. Well, when you consider what we've talked about for the last, I don't know how long I've been on this, but also what we've been talking about for a while now, and what you've read here repeatedly, I hope it makes sense that carbohydrates are, especially monosaccharides, are small let me rephrase that. Monosaccharides are small and can be broken down quickly. Energy can be released very fast. Lipids are bigger, or fats are larger molecules, or longer chains. And take longer to break down. So lipids are great at storing energy, but they take longer to break it down. If you need energy now, you probably want to use a monosaccharide. Luckily, sugars form polymers, which means you have thousands, if not hundreds of thousands, if not whatever, I don't even know the number, of units of carbohydrates, monosaccharides, linked together in a chain in your liver, and plants have them in their and that where you attach them is important, but I'm not trying to specifically draw these in any particular way. I just am trying to emphasize that they're long chains, all right? So, and I mean extremely, extremely long chains, right? So what's cool about that is when you want to break it down, you start to break down the units, and as soon as you split this up, this can be burned quickly, this one can be burned quickly, this one can be burned quickly, and you can throw them into the bloodstream, and they can travel all around the body, and each individual unit can be broken down very quickly. Lipids are much more difficult to manage because they're long chains. They have to be put in little globules called HDL or LDL, and they have to be transported through the bloodstream to different parts of the body. Then they have to be broken. The long chains have to be chopped down. So the example, I mean, something you might think about. It's not quite a great example, but if you think of like a matchstick, how easy to burn is it to break down and use a match to a bunch of kindling to make a fire, versus chopping down a big tree then chopping the tree down into bits, and then using those bits to start your fire. So that's kind of the difference between monosaccharides and lipids, and at this aspect of the biomolecules is that monosaccharides are small units and they can be easily broken out, where lipids are longer chains, or fats are longer chains. Look at the two types of fatty acids below, saturated and unsaturated. What is the difference between the two? You remember in our discussion with saturated and unsaturated, if you look at the carbon and they're all the single bonds, all of them are full, either they're bonded to another carbon or hydrogen, we call that saturated. As soon as you have a missing hydrogen, the carbon is an unsaturated carbon. It's an unsaturated fat. That means the carbon does not have single bonds. They're not bonded. Not all the carbons are bonded to either hydrogen or oxygen. There's a double bond to another carbon, so it's unsaturated. So the difference, as we've discussed in class, is this. Saturated fats are solid fats like animal fats, lard, butter, whereas unsaturated fats are more fluid and form oils such as vegetable oil. Trans fats are plant oils that are artificially solidified to make them suitable for baking purposes, right? So we can use these, we can change these artificially, and we can make these solid at room temperature, even though they're plant oils. In recent years, trans fats have been associated with negative health issues that are not widely used. Explain the simple molecule term that would have to be done to plant oil to transform it into a trans fat. And it sounds like it's something that would have to be nutty to be able to do. But the point would be, you would take this unsaturated and make it saturated. Well, how would you do that? Well, you would do that by adding a hydrogen. If you add a hydrogen there, now you can layer the fats on top. It'll be solid. Well, without it, this unsaturated fat is kinked and will be liquid at room temperature. But if you add a hydrogen, it straightens out, and now you can layer them out. They become solids, like margarine. Margarine used to be something people ate. I don't even know if you can buy it anymore. They would have a lot of trans fat in it. People thought trans fat was healthier than saturated fat. Well, it turned out that's not true. It's horrible for you. It's actually worse. So, in fact, it's the worst fat you can eat. So trans fat is something we don't want to eat. So you're taking vegetable oil and you're adding a hydrogen to it. What would you call that process, just out of curiosity? I'll give you a second and let you think on it. Adding hydrogen to unsaturated fat to make it solid at room temperature, to make it easier for you to use in baking and such. What would you call it? Well, it turns out we call it, I know it's crazy, hydrogenation. Hydrogenated. If an unsaturated fat has hydrogen added to it, it's been hydrogenated, or hydrogenation is the process. So adding hydrogen to an unsaturated fat to make it saturated, so you can use it in places like baking. You can use it to spread on your butter easily, on your bread easily, and it's not saturated fat, but it is saturated fat. It's just been done chemically. So you take vegetable oil and turn it into solid form. And people thought it was safer, but today we know it's not. All right, that's it.